Okay, thanks for introducing me, Essen. So what I will do is I will give a very brief and basic and preliminary introduction into the topic of cognitive models and how can they be utilized together with neural data to make more meaningful inferences about our data from our studies. Okay, so first of all, I will start by providing a very a simple definition. And whenever I speak about cognitive models, I will be referring implicitly to what I mean by this. So formal mathematical descriptions of latent processes which generate the observed data. So basically they're a form of generative models which generate behavior. They're concerned with describing and quantifying the latent processes which go on in our brain, which produce behavior. So you can imagine in the olden days all psychologists debates about the mechanisms of memory or categorization. So there are these implicit latent models which produce behavior. However, there is no reason to restrict this cognitive modeling just to behavior. We can also implement neural data and come up with more meaningful results, and we can also use the neural data to make better models. So let's start with a very simple example which will be taken from my area of expertise, which is decision making. So let's imagine we are concerned with the question about how does time pressure influence decision making. A very simple question. We have a research question. We design an experiment where we manipulate like all the relevant variables. We collect data, both behavioral data and neural data. This can be any kind of neural modality, be it neuroimaging or any other sorts of neural data such as single cell recordings, and then we can perform a myriad of statistical tests and analyses, and finally we come up based on the results of our statistical analyses with some kind of inference. In this case, there are two types of inferences we can make. First of all, the behavioral inference about what happens in the behavior. So in this case, a common finding would be that when we induce time pressure, the responses get faster, people respond faster, but they are less accurate. That's a pretty common finding. And second of all, we can analyze the neural data and come with some kind of brain correlates with what brain areas are involved with processing time pressure or with accuracy or for the decrease in reaction time, all of that. However, if we take a look at this, what you might notice is our inferences are a bit limited because the only things we can infer about are the end products of what we can observe. So basically the reaction times and the accuracies, and that's all we can tell. We cannot say anything about the process, the brain internal mechanics that leads to these changes in behavior. And this is where cognitive models come in. So in this simple diagram, we could assume that this data is generated by some latent process which can be described by a formal cognitive model like this. So in order to go into a bit more detail about this specific example, I will need to take a little bit of detour and give you an example of a very common framework of models used in this type of decision making. So imagine, so this is the accumulation evidence framework where we assume that there's a family of models, but they all have the common assumption that information in the brain is sampled and accumulated for each option, and whenever it reaches a predefined decision threshold, we make a choice. And I'll show you a basic example in terms of a simple dot motion task where you have to say in which direction do the dots on the screen go, and we can imagine that these two bars represent our internal accumulators in the brain. And whenever one of them reaches this threshold, which is the upper boundary, we make a decision and we press the button, for example, left in this case. So all of these models have some set of common parameters, and I'll mention just the basic ones, which are, first of all, there is non-decision time, which represents which represents the time needed for encoding the stimulus. So this is not related to a decision, this is just the time needed to encode in our visual cortex what's going on for the information to come to our visual cortex, and then also combined with motor execution, which is also not related to a decision, it's simply the time after making decision, the time needed to press a button, physically press a button in this case. And then we have the meat of the process, which is the evidence accumulation. So this is the process of accumulating evidence in favor of the one or the other. And here, many of those different models have a bit of different assumptions. That's a bit beside the point. In this case, which I showed you, these two accumulators were quite independent, but they can interact with each other as well. And finally, an important variable is the decision threshold. So it's the upper boundary, which can be high or it can be low. If we have a low threshold, that basically means that we need little evidence to make a choice. In this case, we'll make faster decisions, which will be probably less accurate. If we have a very high decision threshold, that means we need really a lot of information to commit to a choice. And in this case, obviously our decisions will be better. However, they will be not as precise. They will be more precise, but not as fast. Yep. Okay. So let's assume that we use this model to try as a generative model of the decision-making in this particular task. Then we can take this model, fit it to the data, and perform statistical analyses on the parameters of the model, and then come with some inferences. So I'll give you an example from work, a famous work from the field, which is from Birte Forstmann, who is one of the guests here, unfortunately absent today. What her group did, what they did, is they fitted the model to the data, in this case time pressure, and what they found is behaviorally that the thing that changes with time pressure is threshold, meaning people strategically change their threshold when they have to deal with time pressure. And then what they did is they correlated the behavioral parameters with the neural data to find neural correlates of threshold. And what they were able to show that people dynamically adjust the threshold. Oh, sorry, I went too far. Oh, no, it's okay. The people dynamically adjust the threshold by the connectivity between the striatum and the pre-SMA, which the striatum disinhibits pre-SMA, and that's how threshold is adjusted. So as you can see, it's a way more meaningful inference because we can say something about the underlying neural mechanism which drives the changes in behavior which can observe. However, this is not the only type of analysis and inference we can make based on neural data. We can also use models in kind of opposite way, where we can constrain the cognitive models by our data. So basically what this means is that we can use the neural data to make better models. We can use the neural data to constrain the models, and we can do it on different levels of stringency, so to speak. So we can basically for example, make some biologically or neurally guided assumptions about our model, which constrain the model. For example, a classic example from this would also be the leaky cumulative accumulation model, which assumes that, for example, neural firings cannot be negative, obviously, which then constrains the model, meaning some of the model parameters cannot take negative values, and it has also other constraints. You can also be more stringent, and you can also evaluate the parameters of the model on a neural level. So basically, you have a model which assumes that parameters are estimated on the level of the brain. And what that can give you, and then you can again perform a myriad of statistical tests and analyses, and what you can come up with is basically a biologically realistic model of underlying behavior, which in this case would be a goal to build a realistic model. And one of the examples of the more stringent approach is the Wang-Wang neural attractor model, which is kind of similar to drift diffusion model. However, it is more biologically viable because it estimates some of the internal parameters on the level of neurons, of neural firings. Okay, so this is just a brief, that was the brief introduction. There's also a lot of other ways which I unfortunately don't have right now time to cover. I think some of them will be explained by Brandon Turner later on. What I will go through in this talk is an example from our own work, from our lab, of this framework. So I will describe the experiment. If anyone is interested, you can also see the poster on Thursday. Okay, so the background of this study was that we were interested in finding out what happens when people make choices between equal options. So basically options which have very similar or basically equal value. More specifically, what happens when you make choices between two very good options or two not so great options. And our goals here were threefold. First of all, describe what happens on the terms of behavior, then find the underlying cognitive model which best describes this, and finally we wanted to build a model which also includes neural EEG data. What we wanted to do is fit a trial level regression to this model, which was also an evidence accumulation model. So this is the same framework that I already introduced. Okay, so going step by step by point number one, these are the behavioral results, and you can see the behavioral results are pretty strong and pretty clear, meaning that when people make choices between two options which are very good, they respond much faster than when they make choices between two bad options. And as you can see, the time is like, the difference in reaction time is quite striking, actually. What's next is we wanted to fit a model, and for that, we use the accumulation evidence framework. We use a hierarchical method of estimation, and the first step is to select the best fitting model. So we compared models based on different parameters, so different amounts of parameters per condition or per accumulator, and then we selected the best fitting one. Here is based on information criterion. The red bar represents the best fitting model because the lowest score is basically the best. The second step was to see if the best fitting model is actually able to explain the data to a reasonable extent, and what this represents is the histogram represents the distribution of reaction times from the data, and the density curves represent the predicted fit of the model. So this is what the best model would predict. So as you can see, the fit is pretty good. So we have a reasonable trust in this model that it describes this behavior pretty well. And finally is the model inference here. So what differed most greatly in our analysis. So what our analysis showed is that the greatest difference was in the accumulation rate parameter. So which this means that in case of two good options, the speed of accumulating evidence was much faster than between the two bad options, or two medium options for that case. No other parameters of the models were that different. So we have this second piece of inference, which is basically the model-derived inference, that we believe that this difference in behavior is driven by the difference in speed of evidence accumulation. And finally, the final piece of this puzzle is to combine the neural data, which was EEG data, with the model-based analysis to try to come up with some kind of brain correlate of this difference, or maybe a brain correlate of the accumulation rate. And we wanted to do it on a trial-by-trial level. So this, first of all, is our ERP signal averaged across all conditions. And you can see we had like two components which were specifically of interest. So first of all is the P300. P300, it's because it was previously in the literature associated with the speed of evidence accumulation, for example. So we had good evidence to believe that it might be crucial. And the second component is a very early component, is the N1 or N100 component, which which was very prevalent in our data set, and our kind of working hypothesis was that it might signify the end of the encoding phase or the beginning of the accumulation process. So we've set a few different possible hypotheses, like for example that the latency of P300 might be important, or the amplitude of P300 might be important, or which was our kind of best bet, or the most interesting hypothesis at least, that the slope between the two might be related to the accumulation rate. So basically and the slope is defined basically as the amplitude, the difference in amplitude between these two, and divided by different latency. So basically, the idea is that the steeper the slope, the faster it goes from the negative peak of N100 to the positive peak of P300, that this actually drives accumulation, or this is the neural correlate of accumulation, and we wanted to test this. So since we wanted to test this on a single trial level, this was not obvious, both in terms of the EEG and in terms of then the model fitting, and I'll show you why right now. So first of all, the first hurdle is to get a good estimate of a single trial ERP signal. For those of you who work with EEG data know that EEG data from single trials is super noisy. It's very difficult to see any kind of components. So what we did is used a method which is single value decomposition, which is basically multiplying the single trial every single trial EEG signal by corresponding weights, and then estimating a single trial weighted ERP signal, which is still noisy, as you can see on these examples. However, it's much less noisy than not just using the raw data. And in some of the cases, you can see pretty clear examples of N100 and P300. In some of them less. However, it is still a way to boost the signal-to-noise ratio in this. So that's how we estimated these two components on a single trial level, and then we wanted to fit this into our behavioral model, into our accumulation evidence model, which was also not obvious because when you do a standard analysis using evidence accumulation, what you get is you get parameters per condition, for example, per condition. So you get like a distribution which describes the accumulation rate, and you can get the mean and standard deviation. However, you will not get a single trial estimate of drift of accumulation rate. So what we did is an approach quite recently introduced in the literature. Basically, we estimated, we fit a regression equation into the model, and we assume that V, which is the accumulation rate, accumulation speed per trial, is equal to the intercept, which intercept in here signifies basically the mean accumulation speed per that condition, plus the EEG signal of interest, in this case, for example, the slope value multiplied by the beta coefficient, which is the coefficient of the regression. So you can see this is basically a regression equation. And by estimating the value of the beta, we can make inferences whether this parameter actually influences our neural signal and whether it's related. So that was our approach. So what we did then is we fit the models, compared some of them, and as you can see here on a similar diagram than before, that the N1P300 slope model with the regressor was actually the best fitting model. It also outperformed the no regression model. Obviously, this criterion takes into account model complexity as well. So we were able to show that this is the best fitting model. And finally, a final piece of evidence is to see the distribution of the beta parameter, which is the parameter of interest here. And this is the distribution of the beta parameter, posterior distribution of the beta parameter, and we can see that around 97% of the samples of this parameter are above 0, which means that we are 97% kind of confident that that beta is positive, meaning that the slope, the steeper the slope, we are 97% confident that the steeper the slope of the N100, P300 as measured by our method actually is a neural correlate of evidence accumulation. Okay, and that's it for me. I managed to make it in time, fortunately. Oh yeah, let's make a short summary of what I've told you, of what we've learned. So first of all, cognitive models are formal representations of data generating processes, which we can understand them as kind of generative models. Of behavior. And they can enrich our understanding of the neural data. For example, we can use them to look for brain correlates of the underlying neural process. We can also use the neural data to aid our understanding the underlying process, so basically to constrain the model and to understand the neural mechanisms better to make more realistic neural models. And there is a wide variety of ways of combining the two. You can also do kind of both together, like in terms of, for example, joint modeling, which I'm sure Brandon will talk more about. And yeah, there is a lot more that you can do with it. Thank you a lot for your attention, and yeah, thank you.
Okay, thank you, Wojciech. Before moving to the next talk, I would like to remind that at the end of each talk, we will have 5 minutes of question and answer session just for burning questions, but you can always write your questions on the app. And also at the end of the course, we will have 20 minutes of round table. So now I would like to introduce Birte Forstmann from University of Amsterdam and Brandon Turner from the Ohio State University. And the talk is given by Brandon Turner. So please, Brandon.
Hi, I'm in fact not Birte Forstmann, but I'll be filling in for her, so sorry to disappoint. I'll do the best that I can. So I'll be talking about different ways of approaching the analytic process of linking brain to behavior. And so this really starts, at least for me, I mean, you can go back pretty far for this problem, but I sort of draw the line with Davida Teller. So she developed a set of formal axioms that kind of explain what it means to relate a neural physiological measure to a behavioral pattern. And, you know, this was later relaxed by Jeff Shaw, who kind of took this to mean that, you know, we can't really define an axiom, but we can relate it sort of probabilistically, so to define a statistical pattern, basically. And then this was later expanded even further by Birte Forstmann, who made this a probabilistic link. So what she advocated in her paper that really kind of set the stage and motivated a lot of the work that I've done is this really nice figure called the model in the middle figure. And so in this figure, it articulates the many different ways that we can come to understand a cognitive process. So we can, you know, define an experimental psychology. We can define, you know, what the experimental design is, go out and collect some data, fill these cells of the experimental design, and then run some statistical tests to say whether or not the distribution of data are different. In the field of mathematical psychology, you develop a system of mathematical expressions that are regulated by a set of parameters, and these parameters change the effect. So, you know, luckily that's already been introduced. But the goal here is to estimate model parameters and use them as a proxy for understanding some aspect of cognition. And then lastly, we have traditional neurosciences, which at least statistically speaking are very similar to that of experimental psychology. It's just that here, you know, we have better plots, right? They're really colorful brain plots and so on. And so what she really nicely advocated for in this paper was that you can relate the parameters in the cognitive model to neural signatures of interest. And you can do this a variety of different ways. And so just recently, well, actually I'll start with an example just to kind of help motivate why you would want to do this in the first place. So for a standard cognitive model, there are different ways of explaining simple choice response time data. So let's say we have 3 response times, and those are represented here as the x-axis. So this blue, red, and green are supposed to represent those RTs. Now, if you present the exact same stimulus to an observer, what you will undoubtedly find is that the RT varies, right? They don't respond in exactly the same way every time. So there's some variability. And people characterize this variability in a number of different ways, and there are 2 options that I'm presenting here. One is to express the rate of evidence accumulation as being different, and another is to express the total amount of information that you need as being different from trial to trial. Now, there's a problem with this in a very simple term. It's just the Grice representability that was raised by Jafarov back in 1993. And the idea is that whenever you change the variability, you can mimic the RT data perfectly. Okay, so this is just two examples here. So if I have these three RTs, and I have a cognitive model that explains how those RTs emerge by a simple rate of evidence accumulation. So these trajectories are the model, like was already described. They're racing toward a threshold, and whenever you hit that threshold, that's when the RT is described. So basically, the first model here is describing how those RTs emerged by a function of having the drift rate vary from trial to trial. Now, on the right panel, we have exactly the same representation, but now the drift rates are held constant, and the threshold is what's changing. So there's variability in the threshold instead. As you can see, you can still make the exact same account where this black line now is the drift rate, and it's arriving at three different thresholds. Okay, so it arrives at the blue one, the red one, and green one, and as you notice, the RTs match perfectly, right? So this might be a problem, right? If you can't really meaningfully distinguish what these cognitive models parameters are doing in terms of just behavioral data, that could be an issue for interpretation. And so one example, and this is kind of an old example, but I think it's fundamental, I think, to this field or this area of research, comes from Haynes and Shaw. So going back to this drift rate model, let's just focus on that for a moment. We have our three RTs. Now, if you measured and you quantified properties of the model that explain these RTs, you can basically just ask the question. Let's focus on this black line here. So this is the rate at response. So what was the rate of firing at the time of the response? So what we do is we go to these RTs over here, and we just simply calculate what was the total amount of firing that was required to make a choice. Okay, and so as you can see, that's exactly the same, right? The threshold's fixed, so we should expect that the rate of firing, whatever it happens to be, 12 in this case, is going to be flat. So these black dots correspond to those three rates. Okay? Ignore the gray line for now. Now, if we do the exact same thing, but we ask a different question, we say, what's the rate of growth at the time of the RT? So we go back to these RTs and we ask, what was that trajectory rate? What was the slope of that line that eventually produced that choice? Here we see that, the blue is here. It was, basically, the green is here. As the rate goes up, the RT tends to go down. So these black lines all correspond to those three RTs. Okay, so they make different predictions with different questions. Now, if we go back to the threshold model, we can do the exact same analysis and plot those with the gray points. And as you can see here, they're orthogonal, right? So basically, we get two different patterns. And so what we can do is ask, if these trajectories are supposed to represent accumulators in the brain, right? If they're actually some neurophysiological signature of that accumulation process, then we ought to be able to figure out what those patterns look like just from brain data. And so that's exactly what they did in this nice paper. So they went, they took all the RT distributions, they then binned them, okay, according to different RTs. And within those blocks, for trials that ended up in that bin, they calculated certain properties of the neuronal firing. So in one, they calculated the total activation level. In other words, what was the rate at that time, when the choice was made. They also calculated with a simple slope function what the rate of growth was up until that point. Okay, and so as you see here, this is, it's flat here, but it's going, downward in this example. So that would suggest that, the rate of firing is changing from trial to trial, and it's not a threshold mechanism. So this really is what kind of gave rise or justification to a lot of the latent variable modeling that came after that. So there are different ways of linking brains to behavior. This was a very theoretical approach, right? It was sort of data analytic, but it tested different theoretical assumptions by making concrete predictions to neuronal firing. And in this recent review paper, we talked about these different ways, just trying to organize how you can link to brain to behavior and what had been done so far. Now, of course, this is just a simplification. There's a lot of literature out there on this problem. So I'm sorry if you don't fit quite nicely in here, you should tell me so I can reorganize this. But basically, there are two different goals. So if you divide up the different links on the basis of what the researcher was intending to do, this actually creates a nice taxonomy. So in the first block, you can use neural data to constrain your behavioral model. So if you are trying to increase the complexity of the model, but you've kind of run out of run in terms of the behavior, you've captured all of R squared you can think of, you can look to the brain data to help guide further justifications of the model. Another approach is to use the behavioral model to predict neural data. So if you have a behavioral model that you really endorse, and you want to try and look for the latent representation that exists in the brain, you can do it this way. And another one is simultaneous modeling. So if you don't necessarily weight neural data over behavioral data, you want to consider the joint distribution of those two variables. There are a number of approaches for doing that. Okay, so I'm going to walk through each of these blocks and just give you a couple of examples. So just think of this like a highlight reel. So the neural data can constrain the behavioral model. So a lot of times when you're trying to develop a cognitive model, it's a process, right? It's iterative and it practically takes a whole career, right? So you have to develop this model. You make very specific assumptions just to get the model to make predictions for, whatever behavioral metric you're interested in. And rather than just making these choices sort of arbitrarily, you can look to inspiration, right? And if we believe that the brain is really like housing the mind, it's the biological instantiation of it, then it would make sense, right, that we would look to the brain to justify some of these latent cognitive assumptions. And so these are just some examples. I guess I didn't talk about this. So I'm representing the neural data with the orange node here in, and I'm representing behavioral data with this blue node B. And then I'm just kind of arbitrarily defining sets of parameters that describe those distributions with these circular nodes here. And there are different parameters that are sort of meaningless, but basically there are different ways of doing this. And so one is theoretical, right? So you can definitely make a connection to brain data and with actually no statistics at all. You can just look for inspiration and consider biology, anatomy, and things like that. And so there's some great examples of this. Perhaps one of the most powerful in cognitive science is that of the PDP modeling approach, J. McClellan, Rumelhart, and so on, where they consider, and these have grown into things like deep neural nets and so on. Now, one good instantiation of this that was pretty concrete was the leaky competing accumulator model that was already mentioned here. So it basically extended classic models of decision making by asking a couple of questions, making different assumptions here. So one is that the accumulation of evidence may not necessarily be anti-correlated as it's represented in diffusion models. And there are a number of different things. So we know that neurons they exhibit leakage. They also exhibit local and global kinds of inhibition. And so they really took the biology of neurons seriously in developing this cognitive model. And whenever you plot this model, it has these interesting properties. So this is an example of lateral inhibition. So whenever these two accumulators, if you're representing a choice as one of these two accumulators, they're sort of racing to the threshold, as I described earlier. And at some point, a bifurcation ensues, right? At some point, one of these choices starts to gain an advantage, and the so-called rich get richer phenomenon sort of breaks this trajectory. And so this mechanism here, that's known as lateral inhibition. And actually there are people that have identified when that happens in the brain, so that you can relate parameters of that process directly to brain data. Now, there are other approaches like two-stage behavioral. So here you're just using the model to characterize how the implementation level is carried out. So this is really nicely illustrated in the work of Wang and Wang. So they really take the biology very seriously, and they kind of implement various forms that are neurally plausible, really extending the work of O'Sher-McClellan. And I wrote this here, this 7200, so that's like the number of parameters in the model. So these models get really complex, but they exquisitely capture patterns of neuronal firing and choice response time behavior. Other approaches use what's called a directed input. And so here the idea is if I really want to take the neural data seriously, I can just use the neural data to directly replace mechanisms in the cognitive model, right? So one of the most powerful aspects of cognitive models is they're abstract. We don't necessarily know what's going on in these various mechanisms, so we just posit them as ways to kind of soak up variability in the data. And they're meant to be further refined. And so what these guys did is took this very seriously, and they took neuronal firing patterns that were arriving from frontal eye fields, and they used that directly to drive accumulator models, just like the ones I described earlier. And they asked different questions, such as, if I have leakage or lateral inhibition and so on, I want to describe the map from neuronal firing at FEF to eventually a choice and a response time. And so they tested various components of this model and eventually concluded that, a gating mechanism works really well, but I'll lead you to that paper. Okay, so other approaches is to use a behavioral model to predict neural data. So sometimes we have models that really describe the cognitive model very well, but they may not really give us any understanding of what the neural basis of the mechanisms are, right? So we have to kind of figure out what that is. We really believe the model, and we want to look for patterns in neural data that, justify these assumptions, we can use this approach. So here are two different examples. One is just the reverse of what I already described. This one here, though, is a bit interesting. So here you're trying to fit a cognitive model to behavioral data, define what the internal state of the model is through the parameters theta, and then you're trying to make predictions about brain data that you then correlate. Okay? Anything that meaningfully correlates with that prediction from the model's internal state can then be used to kind of substantiate claims that this is maybe where the mechanism is carried out. So one of the best examples of this is the so-called model-based fMRI. So if you think of reinforcement learning models, you can fit a reinforcement learning model to some choice data. Then you're getting an internal state. So you may have heard of something like prediction error. So you're getting a prediction error on every single trial. And this is derived. There's not really a parameter that corresponds to prediction error, right? It's just a model internal state. So if you take that prediction error on every single trial, you convolve it with a hemodynamic response function, you can then create a long time series of data that you can then use to correlate with brain data that shows up in fMRI activity. And so if you do that, then you find these really classic results. And you can also use this approach a little bit more strategically to justify which mechanisms should be appropriate. So, for example, if I have multiple cognitive models and I'm trying to figure out like what's the best realization that shows up in the brain, not just the number of voxels, but what's the best descriptor of the latent state. And so these authors, Michael Mack, Brad Love, and Ali Preston, really took this seriously. So one example comes from categorization. So if you know something about the categorization literature, there's this really hotly contested, it's not really hotly contested anymore, debate between exemplar and prototype models. And so these have different representations of how people categorize things into choices. And so what they did here is they just used a very simple categorization experiment, and they fit both an exemplar and a prototype model to their behavioral data. And what they found is that these models, once optimized with respect to certain parameters, they made equivalent predictions for behavioral data. You could not distinguish them meaningfully from just behavior. So what they did there is, there was only one subject for which you could actually do that green dot there. Okay, so what they did is looked a little deeper. So they used this idea of model decoding. So they generated predictions from each model of what's just called a similarity. So for any given test item, they calculated how similar that item was within the current representation that was evolving through time. And once you have that representation, you can then do the exact same tricks I described before. And so when they did that, they correlated the latent state instead of any given parameter directly to data. They get different patterns. So as you see here on the left panel, you get a nice differentiation between green and blue. Green being the exemplar and blue being prototype, and this ended up being significant. And they did all kinds of other fancy things that I'll leave you to read about, and related these similarity metrics directly to brain data. Really nice stuff. The other approach, the two-stage neural I mentioned, is just really kind of the opposite. And this has already been discussed. Birte Forstmann is one of the pioneers in this area, sort of relating cognitive model parameters directly to patterns that you see in brain data. And this has been most nicely demonstrated in that of the speed-accuracy trade-off in the striatum. Now, the last approach I'll talk about is simultaneous modeling. So if you don't necessarily care about any particular metric, but you want to describe the joint distribution of the two metrics, you can posit this as a statistical inference problem. So I know I have two streams of data. My goal is to figure out what is the map between these that best explains both patterns of data. So you're not taking one metric as being sort of the priority. There are a couple of approaches here. So one is the integrative approach, and one is the joint modeling I'll talk about later this afternoon. So joint modeling is shown here. It's just a simple schematic. This one looks a little different, but really has the same basic strategy. So you're trying to relate these cognitive model parameters through a linking function that's fully probabilistic. So it extends some of the statistical arguments for linking functions described by Shaw that I mentioned earlier. Now, the idea is that you have some activation in some brain regions. I'm just showing one area here, the striatum. And ideally, these brain data will have some time series associated with them. And you can use models to describe the time series. And so there's HRFs for that. And you have a pattern of time series, a way of describing how each stimulus affects neural activity. And we're going to call those parameters delta. Now, in a parallel vein, you can use cognitive models to describe the time series of a latent state the choice mechanism, which is shown here. And ideally, this cognitive model should basically perfectly match the temporal structure that's apparent in your neural data. If you have that, you can describe this pattern of trial-to-trial activity in the cognitive model parameters. Just call them theta. And the last step is just to jointly explain how delta and theta coexist. Now, this is a hierarchical structure. You can do this in a frequentist framework, but as I'll show later today, you can do this in a Bayesian framework quite conveniently. The last approach is that of the integrative approach. So here, this is the most close you can be to jointly describing two different types of neural and behavioral measures. So this, there are a couple of different approaches, like you may have heard of Ningo. Actar is another great example of this that really started explaining cognition way back, in the 70s. And John Anderson has really advanced this line of research now, connecting it to neurophysiology. The basic structure of the model, it looks something like this. Here is there are a bunch of these modules that interact with one another, and the rules defining how this interaction takes place over time. And within each of these modules, there are rules defining how information is processed and eventually sent to other modules. And so in 2007, John Anderson took all of these different modules and made very specific assumptions about where they might be instantiated in the brain. And this is kind of highlighted here with these little voxels. This is just one way of doing this. This is a confirmatory approach, but they also explain how to do this in a exploratory way as well. And so this is just one example of what you might get out from an Actar simulation with some set parameters. These are activations at every moment in time on the x-axis of 4 different modules. As you can see, this time series is being activated multiple different times, and what you can do is use an HRF, and you basically just place a hemodynamic response function at the onset of each of those modular activities. Then you convolve that signal, and you can use it exactly in the same way I described with the model-based fMRI approach. So this is a really nice example of that. Okay, and so, so that's basically it. So there are different ways of doing this, and I've described all of them today. There are many more that are emerging, like this field is growing really rapidly. And so, and there's plenty of space here in this little box for creating new approaches, so please help out. That's all I have. Thanks.
Nobody has any questions, so we can continue. So now I'd like to introduce Jian from Peking University, and he will talk about the reinforcement learning models.
Hi, I'm Jian. Today I'm going to talk about the reinforcement learning and how it can be married to fMRI. As just Brendan briefly mentioned, he sort of gave the leak to my presentation already. Recently, reinforcement learning has really enjoyed a surge in terms of its popularity. And I think one of the major reasons is because the recent rise of AI and machine learning. Just click. Yeah. We live in a sort of rather uncertain world. If everything is quite certain to us, then making decisions is very easy, right? Just pick up the ones that give us the most reward. But we know things are quite complicated. For example, in this case, which direction you should go, really, you have some clues, but the clues are not exact, and you can't just blindly follow the clues to get to where you want. One of the principles to solve this problem is using reinforcement learning. And today I'm going to talk about how reinforcement learning can be combined with the computations in the brain. And one of the classic papers making these suggestions is a paper talking about the dopaminergic systems in the brain. So there are some dopaminergic neurons in the brain that project to different brain areas. And one of the classic work in the 1990s showed that those dopaminergic neurons instead of just signaling reward, it signals something different. This is a paper, now it's a very classic paper. As you can see that from the very beginning, if a monkey gets some type of reward unexpectedly, the midbrain dopaminergic neurons sort of fired. And later on, when this training, there's a CS, a neutral cue is paired with the reward. For extended period of time, the dopaminergic neurons firing patterns shifted to the onset of the CS. But when the reward was delivered, nothing happened to those neurons' activity. Later on, the funny thing is that when the CS was up, but by the time the reward was supposed to be given, but no reward was delivered, you see this decrease of dopaminergic activities. For quite a while, this has been a puzzle in both sort of learning literature and neurophysiological field. Eventually, people come up with this idea that those dopaminergic neurons actually were signaling, instead of reward itself, it was signaling reward prediction errors. And briefly, or simply put, you can define the prediction errors as what you get minus what you expect. If you follow this kind of definition, now the mysterious patterns of dopaminergic neurons is not mysterious anymore. For example, in this case, in the early trials, you didn't expect anything, but you do get a drop of juice or water, and that's why it gives you a positive dopaminergic response. When the training happened for a long time, now you expect something, and you did get something, so the response get canceled out. That's why the reward delivery didn't cause any dopaminergic firings. Same thing happened later. When you expect reward, but you didn't get anything, that's why you have a sort of a negative, which is reflected here by the pause of the neural firings of those dopaminergic neurons. Of course, this is very sort of simple and schematic representation of the dopaminergic neurons. Now we are trying to come up with sort of more dynamic ways of looking at the neuronal patterns that's linked to the dopaminergic pathway, and also how the theoretical foundations of reinforcement learning. Really, depending on how you look at the question, you can slice the reinforcement learning theories into sort of different categories. If you focus on the value iteration process, basically you can divide the reinforcement learning into the very popular temporal difference learning, and also the Q learning. Some of you might be very familiar. The actor-critic, which the prediction error signal can train the actor and critic. That's for the model-free part. And also you have this model-based part, which Brandon just mentioned. I will talk about that later. If you just use the policy iteration, basically the idea is that instead of generating value signals for different options, now you internally represent a policy telling you which option to choose, and then use the prediction errors to directly interrogate this policy, tell you next trial whether you should move away or towards certain action. Okay. Of course, you can get even more complicated by, for example, the hierarchical reinforcement learning, and also this multidimensional reinforcement learning. Those are the classic papers that you might want to refer to if you're interested. Today, for the sake of time and also the sort of popularity, I'm going to talk about there's three types of reinforcement learnings: the TD and Q learning, and also the model-based ones. Since this is a sort of an educational talk, I'm going to briefly mention how, once we have picked a model, how we're going to do with this model. We know we usually perform experiments. We collect some data. The data can be either behavior, choice, reaction time, or physiological. For example, skin conductance, heart rate. Once we have those data on hand and we pick the model, we can do model fitting. And model fitting, in general, there are two sort of approaches. One is called point estimation. in the sort of linear regression field, we already know the least square errors, and it can also be used in more complicated model fittings. But most commonly used method in point estimation is probably the maximum likelihood estimation. Okay, and but the drawback of maximum likelihood estimation is that sometimes when people's behavior, especially individual behavior, there's a lot of variance in it, the maximum likelihood estimation tends to give people pretty absurd parameters. In order to adjust that question, you can try this maximum a posteriori method. Basically, this method is by imposing a prior distribution on the parameters, so that now if you combine or time the prior distribution with the likelihood to generate the posterior distribution of certain probability of certain parameters, you have a nicely constrained parameter space posteriori. If the models also involve some latent variables, you may also want to try expectation maximization for point estimation. This, for example, is very useful in Gaussian mixture models. Recently, there's a trend, especially for the Bayesian lovers. They tend to do this posterior distribution approximations. Out of those, you have Monte Carlo Markov chain, the Metropolis-Hastings method or Gibbs sampling, and then you have this particle filter. Also, if you want to make the model even more complicated, you can do this hierarchical Bayesian estimation. The idea is that for the individual parameters you have on your hand, you assume there's a group sort of distribution of the parameters which governs each individual parameter. For the model comparison, usually this comes when we have sort of different candidate models at hand, and then the problem becomes which model is the best in terms of explaining or for your behavior data. There's some methods that people are commonly using, for example, the AIC criteria, the Akiki information criteria, or the BIC, the Bayesian information criteria. That's for, and also you can do the Bayesian model comparisons. I'm just laying out all the possible sort of methods that you can possibly use if you are interested in this type of model comparisons. Of course, these are not specifically restricted to reinforcement learning modeling process. It's for any sort of behavior modeling. Eventually, once you pick the RL model, once you have this model fitting into your data and generated regressors of interest, for example, in reinforcement learning, typically people generate regressors such as prediction errors and expected values for certain options. And also, for another one of interest might be the vigilance signal, how people sort of are sensitive towards prediction errors for each trial. Once you have those parameters or trial-by-trial variables, what happened is that you extract those trial-by-trial variable and you convolve them with a standard hemodynamic response function to generate a predicted hemodynamic response. And then you go to the brain and search all the hundreds of thousands of voxels and trying to find which voxel gives you the neuronal pattern that looks very similar to the pattern that you just predicted. Okay? Okay, now we're going to talk about, this is just sort of toy examples of 3 different reinforcement learning algorithms. The first one is the temporal difference one. In the temporal difference learning, there are states. You can think of them, the states, as sort of different time points in your trial. In each state, there's a value associated to it. The optimum value or the real value for that state is the discounted values starting from that state to the future until the trial ends or the session ends. Of course, before the experiment starts, nobody knows exactly what the state value is. That's where the temporal difference comes from. Suppose you're starting from this state. Now you start moving to the next state, but on the way there, you received a certain amount of reward. If the reward is accurate, that means your prediction starting from this state is completely equal to what you are going to receive, and starting from the next state, the value of that state. If this is accurate, of course, there's no prediction error. You don't have to do anything. But if the prediction is not accurate, the current estimation of the state value doesn't match what you received from that state on, then you generate a prediction error. Once you have that prediction error, you can calculate the amount of prediction error and use that, going back to update, now the new value of that specific state. Okay? And by doing these iterations, you can fit this model to, for example, people's choice or physiological response data. Probably the sort of the simplest version of the TD model is the delta rule that people are very familiar with. In psychology, people used to call them the Raskol-Wagner model. Idea is that you have expectation, the new expectation equals the old expectation plus prediction error times a constant learning rate. Okay, you can rewrite this equation as the new expectation of certain stimuli or Q equals to a linear combination of the previous value expectation plus the reward you just received. Okay. This is a very sort of classic paper using TD learning in a passive sort of Pavlovian conditioning experiment. The setup is very simple. You have different Qs, but it's secondary conditioning. You start from QA, and most of the chance the QA will lead to QB, and then QB will deterministically lead to a high level of electric shock. But occasionally QA will lead to QD, and vice versa for QC. You know, QC most of the time goes to QD, and QD leads to a low electric shock. But sometimes QC goes to QB. Okay. If you ask human subjects to do this, they learn this task very fast. Based on the paradigm, something that we can do is to generate two types of regressors. One is the so-called the temporal difference value regressor. That is, when the Q comes up, what do you expect? For example, for this first one, the QA comes up. Since QA most of the time leads to QB and then to the high electric shock, so when QA comes up, you know quite sure that you're going to receive a shock. Okay? Suppose that learning already completed. But for QC, which is this guy, you know, once QC shows up, most likely you're not going to get anything. But until QC is switched to QB, now you know that you're going to receive a big shock. Okay? So by fitting the learning models into people's behavior, in this case, skin conductance data, you can generate this type of regressors. This is for the value, state value representation, and these regressors are for the prediction error regressors. And the next thing you can do is just, I mentioned earlier, you can convolve those sort of time series regressors with a canonical hemodynamic response function and go to the brain and look for the neuronal sort of correlates for those regressors. And not surprisingly, as Brenda mentioned, usually if you use the prediction error and you go into the brain, what you find is typically like midbrain or striatum. Of course, you can make the model even more complicated by, because before we were assuming that the learning rate is constant, now you can even make a sort of hybrid model by change the learning rate to a dynamic variable. Now you have both the prediction error as sort of a variable, and then the learning rate as another dynamic variables. And then same thing happens. You go to the brain and look for the learning rate, which now is not a constant parameter for each subject. It varies with trials, and also the prediction error. By fitting the model to people's skin conductance, you can see, you know, usually they get a reasonable fit. And then you go to the brain, you find, prediction error seems to be located in the ventral striatum, and the dynamic learning rate seems to be associated with activities in the human amygdala. Okay, now really quickly jump to the Q-learning part. And the Q-learning basically solves the question of when you have, you still have states in this kind of games, but now you have control. Basically, you can make a decision as to which way to go. For example, in a maze running task, if you're mice, you're standing at the starting point, you can go right. Once you reach state 2, you can decide to go to state 3 or go back to state 1, which is the starting point. This is sort of typical dilemma that a robot is facing. In this case, the robot is really moving in this terrain. Of course, if the robot touches the bomb, then game ends. If it touches the lightning signal, it will collect a certain amount of reward. The final goal is to train the robot so that the robot learns which way or which is the most efficient way to get to the end point. Typically, for this type of experiment or paradigms, we use Q-learnings. The Q-learning is nothing but adding sort of one more dimensions to the temporal difference learning algorithms. In temporal difference learning, the time just the passively goes by. But in Q-learnings, you have a decision to make. You can go either right or down for each option. And the updating rule is that once you, for example, we can assign sort of default values for all these states, and then once you visit a new state, you get to collect what's in that state. For example, you go from here to here, you collect one sort of unit of reward, but also you get to update how good this current state is. That's the idea. And this is sort of the pseudocode for the updating algorithms. Here is just a sort of dummy examples. Suppose that you are in room 2 and your final goal is to get out of the room, which is denoted by number 5 here. Starting from the very beginning, the state here 10125 labels the names or the numbers of different rooms. You can take different actions. For example, if you're in room number 1, just by going to number 5, you exited the room, and then you can artificially assign a big sort of number of reward that encourages people to go from room number 1 to outside. Following the same mechanism we just talked about, you can easily see how this sort of Q-learning model works. For example, if you're starting from state number 1, which is here, if you go out, if you just happen to make a decision to go out, and then you collected a big amount of reward, how big is that? So this is sort of Q-learning tables, right? If you're starting from state 1, you go to 5, you collected 100. And this equation tells you how to update that. The new sort of Q value for that state 1 and 5, at state 1, take action 5 equals to the old value, which is 0, and plus some sort of learning rate times the maximum amount of if you're standing at state 5 and what you're going to do, right? So this will give you updating algorithms. Now the Q value sort of lookup table, instead of all zeros, this is the sort of initial setup. Now you updated the Q value lookup table. Now from state 1, room 1, if you take action 5, which is going out, you're going to be collecting 100 unit of reward. You can do this kind of iterations all continuously until you sort of converge the Q lookup table into some stable values. In this case, you realize that no matter where you start, for example, in this case, you're starting from room 2, going to room 3 will give you, the Q value of going to room 3 will give you 64 unit of reward. And starting from room 3, which is here, you can either go to room 4 and exit from there, or you can go to room 1 and exit from there. Once you train this Q value lookup table well, and then you can follow this trajectory very easy. Pick any state you're at and just look at all the values associated with different actions and pick the ones that give you the biggest state action value. Of course, this prediction error signal can be used, as we mentioned, to update the state values. That's something we did. And also can be used to update action selection, which is sort of policy interrogation, which we will not talk about. Last one, sort of, or the last to the second. This is a simple sort of one-stage experiment. You have two options, which people usually do in their experiment. You press those two options. Every time you select either one on the left or on the right, you receive certain amount of reward, which is represented by the bar height in the middle. You keep doing this until you find, which option is the best one. And typically, solving this type of problems involves Q-learning, right? You assign Q values for each option, and then you update those Q values based on the reward you received. For example, you press this option, you get certain amount of reward, and use that to interrogate or to subtract the previous expectation to generate the prediction error signal, and use that error signal to update your value expectations for both option A and option B. Once you have a fairly or rather accurate representations of the state or action values, and then you can feed them into action generation process. Again, if you do that, you pull out the venture stratum, as people usually do. Lastly, I'm going to talk about the now very popular ideas of the model-based or model-free reinforcement learning. This idea actually is rather straightforward. This idea all comes from this original paper in 2010 by Nathaniel Dahl. It captures this intuition. So, for example, this is a two-stage decision-making or learning problem. At the early stage, or stage 1, you have two cues on the screen. By choosing one of the cues, and that cue will lead you to another two states. Most of the time, it will lead to a state in the green state. 30% of the time, if you choose that cue on the left, it will lead you to another state, the blue state. Vice versa for the option on the right side of the screen. The interesting is this. So once, for example, if you choose this option and landed you here, and then choose one of the options following it, you received a certain amount of reward, or you didn't receive an amount of reward. The question is about how do you assign credit. If you choose this option and you do receive reward, of course, this guy tells you, this is a better option. Next time, I might be more inclined to choose this option. But the interesting thing is not about this; it's about these two. So, model-free reinforcement learning will give you an intuition that, given you choose this one and lead you there, that means if this turns out to be good, then this guy is good, but also this guy is good, right? Because it tells you to go there and you collect a certain amount of reward. But model-based RL will give you a different prediction. This may be more clearly demonstrated here. For example, in the model-free mechanism basically tells you in a rare condition, the red one, if you in a rare condition, for example, you choose this option, most of the time it goes here, but in a rare condition, it goes here. In this condition, if you receive certain amount of reward, so you know this action must be good, but then preceding this action is this one, so this one must be good, right? But we know from a model-based perspective, most of the time, this option doesn't lead you here. It's this option. So there is a direct contradiction between the predictions of the model-free reinforcement learning and the model-based ones. If you just focus on the red bar in the rare condition, and also that red bar in the of the rare condition in the unrewarded situations. So to capture this intuition, people developed this model-based, model-free approach. In the model-free, just as I described earlier, it's just a simple Q-learning. But in this case, remember, in order for you to get to assign credit to the sort of the second order or the higher order of the actions, you need to add an eligibility trace. But that's something else that we can talk about. In a model-based approach, things can be quite different. In this case, you assume people already know the state transition matrix. For example, in this case, somehow people already have a map in their mind. They know that by choosing the options on the left, they have 70% of chance going here and 30% chance going there. Same thing for the other stimuli, which is quite demanding for the subject. In order to calculate the state action values, the subjects or the animals need to do all these calculations online whenever they see a stimulus comes on. That requires a lot of sort of working memory loads. Once you have the model-free and model-based components, what these people did is that they constructed a hybrid model by linearly combining the model-based component and also model-free component and generate a net Q value for each state action pairs. And again, they feed these pairs into this policy algorithms to generate or predict certain actions based on which state or which actions the subject is taking. So that's for the model-based, model-free approach. So, oops. And if you use the prediction errors generated from the model-free approach as well as from the model-based approach, because those two prediction errors, they're sort of uncorrelated with each other. And then if you use them to go to the brain and hunt for their neural correlates, and surprisingly, you find neural activities in the striatum that correlates with both type of prediction errors. This tends to suggest that the processing of both type of prediction errors seems to be sort of co-localized in the same brain areas, meaning that this brain structure is responsible for two types of reinforcement learning mechanisms. So just to wrap up, I hope I have time or have the clarity to tell you about all these three different learning mechanisms using examples. The temporal difference learning using the now the classic papers of the secondary conditioning paper of 2003, and then the Q-learning using the robot example. And if you're interested in the model-based, model-free approach, you should go and read this paper, which is also classic in the 210. Okay, thank you.
I think my slides are lacking. Okay, thank you very much. So hello everyone. I'm going to talk about some model-based assessment of brain-behavioral relationships. I think I'm going to start with a simple question. So typical cognitive neuroimaging works like this. So you do an experiment and then you try to look for brain correlates of whatever experimental manipulation you had. And then the question is, would you or do you have to care about behavior on top of this? So why should you care? And I guess the first answer relates to the fact that you potentially have to compare your results to the gold standard in neuropsychological assessment, namely the behavioral assessment of brain lesion patients. So this is one example of this. So this is a study by Benedetto Di Martino. He published that very well. These are two patients with a bilateral lesion in the amygdala, and he was asking them to perform risky decisions. So essentially, they would have to say yes or no to a bet, a gamble, that was composed of a loss part and a gain part, 50% probability, and he varies the gains and the losses. And what he found was that these patients effectively became insensitive to losses, so they were gambling much more than their controls. And then that's a result already interesting, but in fact he did also an fMRI experiment with the same sort of experiments, the same sort of design, and what he found in the amygdala was slightly different. In fact, the amygdala was responsive or supposedly responsive or supposedly responsible for the framing effect. whether you frame the decision in terms of a gain or a loss. So there is a mismatch between the neuropsychological assessments of these brain-injured patients and the fMRI results in the same context. So if you want to compare these two kind of studies, somehow you probably have to care about behavior. Another example is this is a very dramatic example. So this is a study by neurosurgeons, and what they do is they operate people that have low-grade gliomas, tumors in the brain. And what you see here is this kind of a patient that has a tumor in Broca's area before and after surgery. And then these guys removed Broca's area in about 100 patients. And they could show that 6 months or 1 year later, people actually recovered language. So, and in fact, language was localized in the right prefrontal cortex. So here again, you can think of it as an example of a mismatch between assessment in brain lesion patients and fMRI, typical fMRI assessments of the same sort of functions. And I guess the key here is potentially that the brain has the ability to remap input to output to recover function. And somehow this is probably due to the fact that behavior itself is degenerated, right? So there are many ways for the brain to get to the same behavior. So there's the dual question, right? So why should psychologists care? And I guess one aspect of that question relates again to the fact that there is this form of behavioral generation. So this is a study of synesthetes. Synesthetes are people who associate letters with colors, for example. And this is an fMRI study showing that 2 sorts of synesthesia occur depending on the type of network that underlies this behavior. So there is a degeneracy in the sense that different physiological states on your anatomical circuits can lead to the same phenotype, behavioral phenotype. And of course, neural data can constrain cognitive theories. So this is an example study taken from our group. The design was particularly simple, so people were asked to rate paintings in terms of how much they like on a scale from -10 to 10. And we paid special attention to what happens in the VMPFC, in the ventromedial prefrontal cortex, just before people were actually asked to rate these items. And what we could show was that activity prior to stimulus appearance was predictive of the value ratings. And the reason that's the case is that the VMPFC fluctuates in an ongoing manner, and then that fluctuations adds, or actually the value that is elicited by the stimuli adds on top of these ongoing fluctuations. And because this system is somehow sticky in time, it cannot escape the influence of the pre-stimulus activity, and therefore the post-stimulus value ratings are influenced by the pre-value or pre-stimulus VMPFC activity. So this is a very simple example of how neural data can inform cognitive theories. Right, so what are typical methods that are used so far to relate brain to behavior? So I'm going to take 2 examples, and I think the speakers before me actually gave a much broader picture of the type of methods that can be used. But the first one is effectively decoding of behavioral outcomes, right? This is a very famous study, a bit old now. They did a very simple decision task, and then they used decoding to predict the choice, and they could show that they could predict the choice of the people about 7 seconds before they made the choice. That's impressive in itself. These kind of techniques are very useful to try to make a link between brain fMRI activity and behavior. They do something bizarre though. They forget entirely about the task itself. There is no notion of what is the experimental paradigm that was used to elicit the behavior itself. And this is another type of techniques that has been used widely now, model-based fMRI. So in that example, this is a study from the TNU, the Translational Neural Modeling Unit at ETH in Zurich. So this is essentially, it was framed as a social task, but that's a reinforcement learning task. And then they had this hierarchical reinforcement learning model, and they could extract from that different regressors that would vary across trials: volatility precision, volatility prediction error, advice precision, low-level prediction errors, etc. And each one of these regressors correlates with a different brain system. So that's interesting again. These kind of techniques, though, what they can't do is they can't use neural data to inform, for example, about inter-individual differences. right? So the only thing you can do really is to confirm that the assumed computation that was underlying your behavioral model, your cognitive model, actually you can look in the brain and see and look for evidence of that computation, but you can't use neural data itself to, for example, say whether that computation was slightly different in one individual or another one. Right, so I'm going to describe 3 techniques, very simple ones, admittedly simplistic, that somehow address the question of relating brain and behavior slightly differently. And I guess the focus here is on trying really to make the most out of the fMRI data. I mean, that could be used with other type of neuroimaging techniques, but here I'm going to be using it with fMRI. So let's begin with this, mass univariate mediation analysis. So that's the work of a PhD student who's working with me, Jules Brochard. He's bald, isn't he? Right, so here is the overall picture, right? So that's what we aim at. So somehow you have a psychologically well-defined task where you have incoming information, cues, action outcomes, stuff like this that enter the brain, and that eventually elicit behavioral responses, choice, reaction time, subjective reports, memory success, stuff like this. And this process, this transform from input to output, potentially could be influenced by psychological factors, such as the way you frame the task, the social context, etc., but also by biological factors, such as drugs or lesions. And then, okay, that's the overall picture. We'd have to have an overall picture that somehow tells us how a particular cognitive function is implemented in these terms, I guess. And then, of course, we can't do that with mass univariate mediation analysis, but what we're going to do is that we're going to try to identify brain regions that both respond to inputs and also determine outputs above and beyond what can be said using the experimental paradigm. So we're going to use the fact that the behavior is not entirely predictable from the task factors, and somehow that there is some information in brain data that above and beyond the experimental paradigms can still predict behavior. Right. So here is how it goes. So you begin with a design matrix that contains the task factors in time, right? And then you relate that to your fMRI time series itself. And eventually that fMRI time series has a link to behavior. And you write 2 regression equations. One is a regression that says somehow fMRI activity within one voxel has to correlate to your task factor of interest. And then you write another equation that says your behavior has to correlate with your fMRI time series, but also potentially to the design, to the task factors. And in fact, the correlation between the behavior and the fMRI time series has to be corrected for the task factors. And then you test for a null assumption, which is essentially somehow that the testing that the flow between x and y through M is broken. So it's either A equals 0 or B equals 0. So it's a conjunction null testing. And if you can do that, you can use SPM, and then using a standard mass univariate machinery to get at p-values that are corrected for multiple comparisons in the typical way. In fact, we even, so the equations are messed up from now on. I use math type, but apparently OHBM doesn't. So all the O's there are supposed to be epsilon. They are noise terms, right? So of course, if we do things the way I just said, then we have to do a regression from brain activity to behavior, voxel by voxel. This is very, very time consuming. So we found a very simple trick to not have to do that. And the trick is to reverse the implicit causal assumption behind the second equation and to write the fMRI time series as a function of task factors and the behavior. So effectively you include your behavioral time series in your design matrix, if you will. Then you estimate the parameters like this, and there is a parameter that relates the behavior to fMRI time series, which is D here. And then there is a way to go from D to the original mediation coefficient parameter. So we can effectively estimate this native mediation regression coefficients without having to regress behavior on fMRI time series. That's very handy, because now we can use, again, the whole SPM machinery to do the whole thing very quickly, very efficiently. So just one note on statistical power. So the null, again, is a conjunction null, right? So effectively, you're asking, so the typical thing you'd use in random field theory, for example, is that you use the minimum statistics between your two t values, or f scores. That corresponds to the significance of the a or the b coefficients. And in fact, the role of the noise, the noise that actually affects or drives fMRI time series on top of task factors, the thing that actually helps you distinguishing or identifying regions in the brain that actually contributes to behavior above and beyond task factors itself, play a slightly non-trivial role. And in fact, what we can show is that when you vary this amount of noise, the statistical efficiencies, so typically the detection rate, the true positive rate, has this inverted U-shape function. So somehow the noise cannot be too small, because if it's too small, then your fMRI time series and your task factors are too close to each other and there is no information you can extract. It cannot be too big neither, because otherwise you won't find any link between your task factors and your fMRI time series. So it has to be a sweet spot somehow. You can't detect all mediators in there. It has to be somehow in the middle of the mediation chain, if you will. So this is a very simple proof of concept example of this technique in a gambling task, very, very similar to the one I've shown with the amygdala patients. So it's a very simple task. So people are asked to gamble or to accept or reject a gamble, again, which is like 50% chance of here winning 12 euros, 50% chance of losing 14 euros, yes or no. And then you vary losses and you vary gains, and then you effectively ask and people to choose. And this is a typical behavior-only analysis, right? So you use a very simple utility theory-like model where you have a softmax function that describes the probability of gambling as a function of the loss and the reward in there. And then what you see here is effectively that, yes, this model can capture the behavior, but of course there are choices that are not well captured by this sigmoid function, right? So there are choices that go against the average behavior of the people. So sometimes people, for the same kind of choice, they accept or they reject the gamble. So there are, if you will, preference reversals in there. And that we can exploit using mediation analysis. So we run that mediation analysis, and this is the typical classes we find. So they're all corrected for multiple comparisons. And you find some aspect of the brain value system, but there are key regions missing in there. For example, there is no ventromedial prefrontal cortex in this kind of task, or at least not using this data set. So, okay, that's all beautiful, that's all good and well, but can we find some assessment of whether that holds? You know, can we cross-validate that? So what we did is that we took another data set that was using exactly the same task, and then we actually asked whether we could predict behavior of the people in this other data set in these regions that we had identified with the mass univariate mediation analysis, and we compared it to control brain regions. And what you see here is the correct prediction rates using, again, this is now using decoding, so this is using multivariate pattern analysis. And what you see is that effectively there are at least two regions that work better than the usual behavior-only model to predict the choices. So there is evidence that the regions we detect with the mass univariate mediation analysis actually tell us something more about behavior than without. But that's effectively all you can do with a mediation analysis. So you can potentially, using that, again, detect regions that somehow, well, I'm going to come back to that later. So second technique, biologically constrained neural nets. So, okay, that's the picture, the overall aim. And now what we're going to try to do is we're going to try to account for biological constraints on the brain's information processing. So said like this is very grandiose, but effectively you'll see that we reduce it down to very simple questions. So we're going to try to extract evidence for potentially behaviorally degenerate biological constraints from local multivariate brain activity. So what do I mean by this? So this is a typical artificial neural network, right? So you've got inputs in there. These inputs could be in my gambling task. It could be the loss and the rewards in the bet. And then you got hidden layers that pass this information from one layer to the next one, and they reduce down to the behavioral outputs, which would be, in the case of that task, whether the people accept or reject the gamble. In each of these units, you got some activation function here. And then the first constraint we're going to, well, okay, let me describe this function. So we took a Gaussian. We can use other types of activation function, but effectively this Gaussian has two parameters. So by Gaussian here, I mean that the way each unit responds to whatever enters the unit is a Gaussian-like response, right? So it has a mean and a standard deviation, and I'm going to assume that this standard deviation actually changes over trials, because actually it follows range adaptation. So range adaptation, in that sense, it simply means that if the input somehow is beyond the typical scale within which the unit responds, then the scale of that response unit will increase. So there is a learning rule, if you will, that learns nothing except but the range within which the neural unit responds. On top of that, I'm going to use another constraint, which is effectively Hebbian plasticity. So the fire together, wire together kind of rule. So the idea here is that the weights will be modified, effectively increased between two units that are activated by the same input, with a threshold that make it either positive or negative. But it's effectively a fire together, wire together rule in there. So again, this thing learns nothing, right? It's just a hardwired biological constraints that we put on top of the the artificial neural network. So what do we do then? We use essentially representation similarity analysis. So we fit this model to behavioral choices. At each trial, we extract patterns of activity on the units, and then we measure somehow a similarity matrix between each and every trial in the task. And then we go in the fMRI, and then we take effectively regions in the brain. They have patterns of activity in each trial, and we compute the similarity matrix. And then effectively we correlate these two similarity matrices, right? That's the idea. That's RSA. That's representational similarity analysis. And so when we do this with our task, well, first, what the behavior looks like. So again, so this is our task, right? This is the same task, the same gambling task as before, the one I used for the mediation analysis. And what you see here again, so is that we're going to try to capture this unpredictable variability for difficult choices using this kind of like hardwired biological constraints. And when you look at effectively how we can predict these choices that are difficult, we can actually predict very well, we can capture very well these unexplainable choices using these biological constraints. But fair enough, but what you can see as well is that either range adaptation or HAB or a combination of the two actually leads to the same kind of like prediction accuracy gain. So again, from behavior alone, we can't tell which of these constraints is really necessary to explain this behavior. So RSI is useful for that. So this is the typical for one participant similarity matrices that you get from having no constraints, range adaptation, HAB, or the combination of the two. And we took our regions we had identified with the mediation analysis to quantify effectively how well these similarity matrices represents or predicts the similarity matrices of fMRI data. And what you can say now is that the Hebbian plasticity learning, sorry, not learning, but like biological constraint, seems to be most representative of what's going on in the structure of the fMRI data. So again, there is no, so this is just cross-validation, right? So fit the model to behavior, all these constraints, they explain the same type of behavior, look in the fMRI data, okay, looks like HAB more than range adaptation. This is just a proof of concept. So the last thing I want to talk about is dynamic causal modeling of brain behavior relationship. So this is again pushing the, so this is another young fellow working on this, Lionel Rigaud. He used to be a postdoc of mine 4 years ago. He's now an independent PI. He works in Germany. And he's not bald. Okay, so what we're trying to do here is to decompose the relative contribution of brain regions and their interactions to the behavioral response, right? So again, that's going to be clearer in 1 second. And then the overall arching goal here is try to get a model that works at the whole brain level, at the network level, so that we can potentially identify something like a functional degeneracy, right? So that we can predict post-lesional functional recovery. We're not there yet, but I guess these kind of techniques can do that if pushed enough. So this is the core of DCM for fMRI, right? So you have a system of equations that describes neural dynamics within regions of interest. And the neural dynamics are very simple. In fact, they are kind of agnostic. It's just saying, okay, there is some evolution equation that drives this neural system, and we're going to use a second-order Taylor approximation to the evolution function. And then we get terms that we can interpret. So effectively, a term that talks about connectivity at rest. There is something that effectively captures the notion that inputs drive the system. There is some input modulatory effects, such that the inputs can change the connection weight within the network. And there is some quadratic gating effects, whereby activity within one region in the brain can change and modulate connections between two regions. That's DCM for fMRI, or actually the classical version of it. Nothing fancy here. On top of that, you got a balloon model that actually relates neural dynamics to the Bolt signal. So that's much more biophysically informed. I'm not going to go into that. Effectively, it looks complex, but what it does is it convolves the neural dynamics with an HRF that is weakly nonlinear. But it's phrased in terms of a dynamical system. And what do we do to do this behavioral DCM? Well, effectively, we just write down a mapping from neuronal states to behavior. And then that mapping, when these states are, for example, binary, when there are choices, it takes very simple forms, such as a sigmoidal relationship. I'm going to have to reverse that. So we say, okay, we don't know the form of this mapping, so we're going to use very simple assumptions. For example, the impact of neuronal activity right now on behavior is going to be higher than the impact of neuronal activity just before in time, and even before. So that's kind of an exponentially decaying impact in time. That we can write with a very simple dynamical equations. And on top of that, we're going to say the mapping from all the neuronal states that compose the network to the behavioral output, which is passed through this exponentially decaying temporal kernel. That mapping, again, we don't know what it is, so we're going to use, again, a second-order Taylor expansion. So there are first-order term that talks about the way one region drives the behavior. You have bilinear term that says maybe there is some experimental manipulation that changes the way a region influences the behavior. It could be a psychological manipulation, for example, and gating effects of the same sort than we used for the neural dynamics. Then we use variational Bayes inference. I'm not going to go into that, but effectively it's an approximate inference machinery for nonlinear generative models. It's a very robust technique. It's been used now for about 20 years. And then, okay, this is my proof of concept example of the technique. So this is not gambling. This is a very simple, probably the dumpiest experiment you can make. It's a go-no-go task. So people are asked to prepare a motor movement on the left hand or on the right hand, and sometimes we ask them to inhibit their movement, to not do it. And when you do the fMRI analysis, this is what you find. So execution left-right hand movement corresponds to the contralateral primary motor cortex. The preparation instruction is again lateralized, so prepare left hands activates the right premotor cortex, prepare right hands activates the left premotor cortex. And the actual withhold the movement instruction, so the no-go instruction, is not lateralized, but effectively it's in the prefrontal cortex, left and right. People say DLPFC, it's slightly border, but effectively we care about these two clusters here. So we have a network that has two motor cortices, primary motor cortices, two premotor cortices, and two prefrontal regions that are responsive to the task. And now what we want to do is somehow have a scenario about how these regions talk to each other such that inhibitory control is implemented in the brain. So we do effectively what we do with DCM, so we do a Bayesian model comparison, so we have all sorts of models. I'm not going to go into the details here, but we have scenarios that are differ in terms of, for example, how the execution of the movement is translated in network activity. We have different scenarios about how the no-go instruction changes activity in the network. Potentially it could be through an inhibition from the DLPFC to the motor cortices, or in that example, which I'm highlighting here because that's the winning mechanism here, it's the DLPFC that gets off the connection from the premotor cortex to the primary motor cortex. And we had different potential mapping from neuronal activity to the behavior itself. And here there was again proof of concept because we knew this is motor action, so that mapping should be just contralateral primary motor cortex for each hand movement. So there was a winning model. That's the one I'm highlighting here. So the execution effective instruction increases the connection from the premotor cortex to the motor cortex, ipsilateral. The no-go instruction, again, gates off the connection from that premotor cortex to the primary motor cortex, and the execution itself goes from contralateral primary motor cortex. So this is how a typical trial would go. Now, just looking in the, we've inverted the model, we just look at its prediction as a trial unfolds. So when we ask people to prepare a left-hand movement, what's going on is that, so this is primary motor cortex, sorry, this is premotor cortex right, premotor cortex left, this is DLPFC left, right, M1 left, right. We ask people to prepare a left-hand movement, activity rises in the contralateral premotor cortex. If we ask them to execute the movement, then that cortex drives activity in M1, which eventually triggers the action. In case when we have a good no-go trials, what's going on is that both DLPFC lights up, and they are inhibiting the connection from the premotor to the M1, which stops the action when that inhibition is efficient. So that's the scenario we get with the PDCM, but that's not the whole story. So what we could do, for example, is that, forget about the left here. We could show, for example, that we now could predict out-of-sample behavior using this technique. And we could show that we could do much better than decoding techniques. And there's a very trivial reason for that. We cared about the experimental paradigm. That gave us a lot of insight. I mean, that gave a lot of edge for the technique to predict the behavior. But then what we did is that we did a artificial lesion analysis. So, so this is I'm going to show you plots that show the response rates on left and right hand side as I lesion the network, one region after the other. And I'm going to show you different types of mistakes. So this here is what happens when you lesion the M1 on the left side. These are right-handed people. So effectively what you see is that you raise any response on the right hand, nothing happens here, that's okay. But on top of that, you also create false responses on the wrong hand. And that's due to hand dominance effects, due to the fact that the M1 talk to each other through the premotor cortex in there. When you lesion the right M1, you don't get that. You only get no left hand response. And you, of course, leave intact the right hand movements. When you lesion premotor cortices, what you find is that there is no hand dominance effect, and again, effectively the system becomes insensitive to the preparation instruction, so you erase motor production contralaterally. And the most interesting result is what's going on when you lesion either left or right DLPFC. When you lesion the left DLPFC, what happens is that under the assumption of the models, you have some failed inhibition on the left side. But effectively, there is no false positive mistakes on the right side. So disruption of inhibitory control is only partial here. But when your lesion write the LPFC, then under the assumption of that model and under this fMRI data, you effectively disrupt all inhibitory control on both sides. And that we could check whether that was the case, because there's about 20 years of literature on brain lesion patients about inhibitory control. And effectively, apparently, from that literature, it's the right DLPFC that is the most critical. So we had a cross-validated results for that particular fMRI that gave some evidence that this technique could be powerful to detect, well, first of all, the criticality of the regions, and second of all, asymmetries that was not visible from the simple activation studies. So in the map that I showed you before, there is no difference, right? So right and left TLPFC are activated by the instruction of withhold your response. So that's it. I'm done. I think I should have been done already 2 minutes ago. So I'm going to stop now. And thank you for your attention. This is just the two people who really worked hard to get that working. And a bit of advertising of the VBA toolbox. All these tools are in there. You can have a look. Thank you very much.
Now, Brandon is coming back to give us a hand-on session, and then you managed to come here, or you are going to do from there? From here, okay. So.
So I'm going to do the slides first, if you can load those, please. Okay, so for this session, we're going to do a tutorial. I guess I have 50 minutes, so I'm going to go through the slides. There's a paper and associated code with this, so if you have your laptop and you want to download this, you can do that. And then toward the end of the session, you can play along if you like. All right, so earlier I showed this figure, which kind of characterizes the joint modeling process. There's nothing too special about this. It's purely correlational and it's not causal, as we just heard from Jian. The basic idea: you have a brain region. It's going to produce some pattern of activities, let's say fMRI data, but it can also be linked to EEG, and we've done that in other lines of work. We've also connected both EEG and fMRI in the same behavioral model. But the basic idea is, when a stimulus is presented, it will produce some kind of activation. We need to measure what that degree of activation is. That's going to be measured by a beta weight or a delta parameter on every single trial. And with that mathematical description, you can produce a prediction for what you should see in the fMRI time series. And that's going to be important because that's going to be defining the objective function that the Bayesian sampler is going to be sampling with respect to. So once you have this big set of delta parameters for every trial and every brain region, if you're doing this in a multivariate way, then you can turn to the cognitive model. And this takes some work, so there are lots of good cognitive models. If you go to a conference like the Math Psych conference, they develop these types of things that describe choice and response time and confidence and all kinds of decisions. But the basic idea is you can use these types of models to produce latent states over time, and you can lock those basically to the same experimental design pattern that you have that's articulating the bold response above. So then you have a set of model parameters that are varying from trial to trial, and that's going to be represented as theta. Theta can be a big vector. You can have multiple parameters that are varying across time, but they should have some common statistical structure from one trial to the next. And there are good ways of specifying that. So this will all become clear. This is abstract at the moment, but eventually I'll tell you how to do that. All right, so the last step is just to specify how those trial-to-trial parameters should evolve. So if I see a large increase in, let's say, striatum, what does that mean in terms of the cognitive model parameters? And you don't really have to specify that. You can be rather agnostic, right? You can just say, well, I don't know, they're regressed. They're linearly related, and that's all I know. You figure out what the slope is, and the algorithm will do that for you. You can also specify something like a multivariate Gaussian, so they are still linear, but now I can build in bivariate patterns that might exist between brain regions and patterns in the cognitive model parameters. All right, so that all sounds good, but one thing that I can't go back. Yeah, I can go back. Okay. So one thing that I didn't mention is, why would you do this in the first place? Why, it's so much easier just to, fit a behavioral model, analyze my fMRI data, and just press, COR in R and just correlate and just see what that is. There is some good reason for that, and that is this induced reciprocity that's inherited from a hierarchical structure of a model. And the idea there is that information that's contained in the fMRI time series actually affects all of these parameters, which affects the inference structure on omega, and that structure then induces changes in theta. So that means that what you observe in the brain data can actually be used to control and constrain your cognitive model parameters. So you actually get better ability to kind of expand the cognitive model to explain better patterns of behavior and so on. And this actually happens the other way around. So, everyone complains that fMRI data is noisy and it takes all these fancy decoding methods and so on, but if you just fit it hierarchically, or if you connect, as Jian mentioned, if you just connect to the experimental paradigm a little better, you can actually use cognitive model parameters in the model itself to drive the analysis of neural data. And so by, defining this joint objective function, that's why it's called joint modeling, it's because you're specifying two likelihoods essentially here. And the product of those two likelihoods gives you all the parameters that you need to build a full cognitive model. All right, so that sounds great, but it's hierarchical. So just, surveying the audience, who here has fit a hierarchical model before? So, I don't know, maybe 15% or so. So yeah, it's non-trivial, right? It takes some work. And if you're a frequentist, it's really difficult because you have to integrate over all the variability and the single trial parameters to estimate the hyperstructure. But if you're Bayesian, this is actually fairly routine. I mean, we get in the habit of building these hierarchical models, and there are great statistical software programs now where you just kind of specify the syntax, and you don't need to know all the clever algorithms that are behind the scenes. You can just tell, the computer, like JAGS or STAN, this is the structure of the model, and I just basically write code that looks like this, and you just press go, and JAGS or STAN will just take care of the rest. So that's what I'm going to show you how to do today. So it's actually not that difficult. Like many papers we've submitted, that's like the common reviewer complaint is, oh, this is too fancy and whatever, but it's actually not that hard once you get the syntax in. So the tutorial paper, it walks you through that. It gives you some really ridiculously silly examples, but they're for the purpose of pedagogy. And then there's a more realistic example at the end. So if you're interested in checking that out. All right, so the basic components of joint models. You start on one side or the other, so let's just start with the neural submodel. There are lots of ways of doing this, and I told you about how to do, an fMRI response that you convolve, right? So you basically take the hemodynamic response function, the HRF, and you place that. If you're doing a finite impulse response or block design, you'll just convolve that with the experimental design matrix that you have. So you just place one of those, and you'll just add them up. So this is called the linear time invariant property. And once you have that added up, then that explains the entire time series. If you want to start simpler, though, you can use just a simple kind of ANOVA-like structure. And so you can start, fMRI data itself doesn't mean much. It's all about relativity here. So you just start with some, arbitrary distinction of, what the experimental variable is, some basis function, and you just measure what the degree of difference is. So these delta parameters would tell you how much of a change in activity did you observe. Now that change in activity, that's what you can relate to your cognitive model, right? So if changes are important in bold response, they can be related to cognitive models. And these types of ANOVA structures are really interesting if you're trying to do a bunch of arbitrary links. If you have some EEG data and fMRI data and a behavioral model, and you're like, okay, I want to fit all of this, you can do that. You can do that fairly agnostically with these sort of ANOVA-like structures. Now, if you want to get really close to the data, there are people that work really hard on this, right? So there's some types of models for specific tasks or specific stimuli that define what the change in fMRI response should be as a function of the experimental design. So for example, if you have a contrast gradient stimuli and you increase the contrast of that stimulus, it should cause a very predictable pattern in the bold response. And so you have a very nice mathematical description of what you should expect from the experimental design. And you can use that then to kind of establish what the trial by trial activity should be in your bold response. So that's the basic structure. There are other ones that I'm not mentioning here. So another cool one is the topographic latent source analysis. So in that, you'll observe some pattern of activity that's either spatial or temporal, and you basically, have a finite mixture of Gaussians just sitting on top of one another. And with that, you can kind of change, the strength of those Gaussians and the heights of them to create all kinds of complex patterns. And those can be used also to describe, the neural data. So once you have the neural data specified, you need to move to the behavioral part. So as I mentioned earlier here, you've got these cognitive models. There are a bunch of them out there. You can do this, again, fairly agnostically. There's a whole field called psychometrics that does this, where they're not really connected directly to like a process account of a decision, but they can still be used to relate the experimental design variables to the choice that you're observing. And so those are great. The other ones, if you want to get more process-oriented and like test cognitive theories, you have to program those up. Those are a little more difficult. So things like the diffusion model, if you've ever heard of the drift diffusion model, you can fit that within, the custom algorithms that I mentioned before, like JAGS and STAM, but someone has to do that work. And so one person like Joachim van der Kerkhoff, he's programmed that up so it facilitates it. So a lot of these process models are available. You may be familiar with other process models that are analytically intractable. So sometimes you have a model that you don't know what the prediction for choice behavior is. You can simulate it, like you can program it up, and it will generate predictions for data, but it doesn't necessarily have what's called a likelihood function, okay? So like the ACT-R example I mentioned earlier, that doesn't have a likelihood function. It's just too complex, and it's all these dynamics. But it turns out that people have worked on this problem as well. So there are methods, from my lab called the approximate Bayesian computation, and you can mix those types of inference strategies with variational Bayes as well. And so those are very handy. Okay, so that's all there is to say about that. So now we can talk about the linking function. So you have brain data, you have a behavioral model. Once you pick it, how do you relate these two things together? And I mentioned earlier there are a lot of these different approaches. One is just a simple directed approach. So you just specify there's some function, just call it M, that the behavioral parameters are dictated by. So whatever changes we observe in delta, it should produce a very concrete prediction for a behavioral model parameter. So for example, this could be just a linear regression, right? So we just have delta times x plus some y-intercept term, beta, or something like that. So the goal here would just be to estimate the parameters of that linking function. And so again, there are statistical software programs for doing that. So one of them, the HDDM package. So if you want to fit the diffusion model choice response time and you want to regress trial by trial level parameters in a univariate way to neural data, you can just download HDDM, learn how to use that, and it facilitates that for you. And the code I'm going to show you later today also does this, but in JAGS, not Python. So there's just some differences there. So what are the strengths and advantages of this approach? Well, first off, it's really parsimonious, right? It's a very low dimensional solution. The complexity of the linking function scales linearly as a function of increases in neural data. So if you want to add a couple more ROIs or a couple hundred ROIs or even go down to the voxel level, you can do that in this approach. It becomes very difficult if you're going to do MCMC. So for that, if you still want to stay Bayesian, you pretty much have to go to something like variational Bayes to get that done. Okay. One of the, a couple of the disadvantages, it assumes this very specific functional forms. It's not very flexible. If you have some interesting nonlinearities that happen in the brain behavior connection, you're going to miss, right? You're going to misfit. So you have to kind of work through those types of questions. It's also vulnerable to missing data. So if you have some wild fluctuation, so head movement, for example, is a concern here. If you don't really, correct for that sort of thing, and you just kind of feed the time series in there, it won't solve all of your problems, right? So you have to kind of be really careful about that. It can really change the slope, just like in every standard regression problem, right? It's really sensitive outliers, so that's another limitation. All right, so another type of linking function is what we've been using, which is this multivariate Gaussian. This gives you a couple of interesting advantages, and so the idea here is you just specify that there's a joint distribution over theta and delta. So both of them are parameters, and we care about both of their fluctuations, their variances, and their estimates, and so on. And so you can specify that really simply. So here, the m is a function of these omega terms, but omega is actually 2 parameters. So omega is a vector of means, so it would specify the mean of theta and the mean of delta. And again, theta and delta can be vectors themselves. So this could just be a really long vector that specifies the means of all of your parameters. And then omega, the other part of it is the sigma, the variance covariance matrix. So sigma can be completely full rank, and what that means is that I want to test every possible pairwise correlation between every theta and every delta, right? And that's hard. That's a difficult problem. But you can, if you know nothing about how the brain should produce behavior in this context, it's a good idea to just start there, right? That's an explorative kind of approach. Now, this is the benefit of using this approach is that, if you know something very specific about a brain region connecting to a model parameter, you can go into the variance covariance matrix and zero out elements that you don't want to estimate. So you just constrain them to be 0. Now that takes some time to code through and zero out every single thing, but that can be strongly, that can be a huge advantage if you're doing this analysis. All right, so advantages, disadvantages. It's less vulnerable to extraordinary cases, so it's not going to correct head movement or anything, but if you do have some wild outlier in your data, that will just be absorbed, right? Because now you're not directly connecting the model parameters. They're filtered, okay? They're filtered through this mu and sigma that I mentioned earlier. It's also got this nice property of being multivariate. So if you just kind of regress ROI activity onto cognitive model parameters, you might be missing important functional connectivity things that are happening. And so if you have a full matrix that's explaining how every ROI is interacting with one another, that gives you a little bit better predictive power. And this shows up in basically all of our applications. Okay, so lastly, it's computationally complex. There's this big issue of now, I said earlier the complexity of the linking functions scale linearly, and that was great. But here, if you increase delta, like you add one more ROI, it has a quadratic effect on the complexity within omega. This is a problem, right? So that means if you just add a couple more ROIs, it starts exploding, right? It's really difficult to fit those models. And you can't go to the multivariate. At least I haven't been able to do that for voxel level. So that's just too complex. But there are good strategies, and people are developing better and better ways of doing this. So we have these other papers like factor analytic linking functions. So you can just say that there's some structural factor that's going over this variance covariance matrix. And if you do that, you work through the math, it turns out that that's also a linear complexity function. So you can get back to the same regime that you were in with the direct linking function with this complexity. So I'm going into maybe too many details, but I'm just saying that there are efforts toward this. So if this is not quite suiting your interest, you're trying to scale to really high dimensions, there are some methods for doing that. Okay, so the application here is this contrast discrimination task. So first I'll describe the task, then I'll talk about the model structure and how you would do this within JAGS. And yeah, a couple of results from a really simple experiment we ran in my lab. So all of the data can be, or the code and the data can be downloaded here at OSF. We also have it on GitHub. Just go to my website, you can go directly to this. I seem to have lost some things in here. I don't know if these slides are going to be available, but just email if you're interested. But this white text here is actually supposed to tell you the link. You can just Google, that's also appropriate here. So you need JAGS, you need this R package JAGS. If you work in R, you need R JAGS. So R JAGS is just connecting you to JAGS from R. So it's just like one line syntax. You can just say go, basically. Then you need your JAGS. This is called the Wiener module. So Wiener is the statistician that is sitting behind the diffusion model. And so basically they just program it up that density. And once you have that density programmed up in C, you can just directly insert it into JAGS. Any density function, you have to write it yourself, but JAGS can do this for you. All right, and also if you're a MATLAB user, they have things like MATJAGS. They have like ways of connecting MATLAB to JAGS, but that's not my native environment, so I can't really be very helpful in advising you there. So contrast discrimination task, this is the basic setup. Really simple, it's just a fixation point, and then we're going to present some Gabor-looking patch, and we're asking observers to tell us which of two contrasts have the highest, or which of two stimuli have the highest contrast. So the first one's presented, and then the second one comes a little later. These are flickering, just to kind of help evoke some neural response so we can really measure it. And then finally, they just make a choice, and this is much later. Okay, so that's the basic paradigm. Now, we have neural data, and we've already processed that and cleaned that for you. So if you just download that our data set, it will have all of this stuff contained. This is for ROI. This is V1. This is Roman area 17. We just use some mask to define that, and signals from all these voxels are just averaged to produce one neural time series, just to keep the problem really simple. Okay. So if you go to application dataset.rdata, all of that's contained in there. You can load it there. The code that's associated with that is, all the code that's associated has _ application. So here's the basic setup. This is that function I keep mentioning, never gave you an equation. So this is the href.convolve function. So basically, it specifies as a function of time what the degree of predicted bold response should be. And it's sensitive to a couple of parameters. There's a beta parameter, which you see there. There's an onset, like the stimulus onset. And then there are a couple of fixed parameters, which, as you might know, like the href has a sort of canonical form. And those are just pre-specified. They're already loaded for you. You can manipulate that. If you want to actually estimate this, you can estimate it. But, in our investigations, we quickly ran into some identifiability issues. So, just be careful there. But eventually, it will give you that response. Okay, so the other pieces you need are the RJAGs that I mentioned, and then this Wiener module. Now, this is sort of non-trivial to incorporate, this Wiener module. So you have to go do all these downloading things, and if you're not familiar with Linux or Unix shells, it's a little tricky. But luckily, there's this tutorial paper by Joachim van der Kerkhoff that I mentioned earlier, which shows you how to do that. And they're just like step-by-step instructions of how to download this, install it on your machine, and get it going. So that should be manageable. It's not easy, but it's manageable. Okay, so then you can process behavioral data, and you can do all of that within here. So the first thing, what JAGS is basically expecting, if you have choice response time data, is a single vector. Well, I have two, right? Choice and response time. So how do you convert that? Well, the trick is just to change what the RTs mean. So a positive RT means one choice, a negative RT means a different choice. And so this is a really simple trick that you can then give this Wiener module just a single vector, right? And so that once you have that specified, which is shown there, then you can construct your data matrix. So that's what's going on there. All right, so assumptions for the neural computation. I said we're going to use this function, but I didn't really say how, and also it's a decision among two stimuli, right? So the idea is, let's say the first stimulus is presented here. It's of relatively low contrast. All you do is just go up into this function, and you're evaluating what that value is, and that's going to tell us what beta 1 should be. So that's the predicted neural response. Similarly, with stimulus 2, we go up into this function, and we'll get a predicted neural response, beta 2. These two are the neural parameters, right? So it's the difference between these two that we're going to be using to describe the pattern of activity, which is going to ultimately predict us our choice. So we're going to do this on every trial, and we're going to specify the model accordingly. So anytime you see those two parameters, that's what they're referring to, is this slide here. Yep, said that. All right, so the neural submodel, we have this general linear model. The basic idea is, you have a couple of stimulus onsets that are showing up. You take the href that I specified before, that code, and you just change what time the onset was, okay? So the onset, here, I'm just roughing this, it's like 0 and 5 and 9, right? Those are the first three. So you'll specify 3 of those, okay? And so then you can draw this href, and it'll have different betas in this case, and then you'll have to convolve that. Now we have the convolution code also in JAGS, so we just do that within the syntax for JAGS, so that's really helpful. It is just a finite impulse convolution, so if you're into these details, we're not doing boxcar. That's a little bit more difficult. We have a follow-up paper, though, if you're interested in that does that within JAGS as well. It's just not in the tutorial. All right, so yeah, this paper here by Fiona Malloy, one of my students, did the boxcar convolution in that paper, so if you're curious about that. All right, so specifying the GLM, we just do it this way. So we have these different onsets, and they're controlled by beta parameters. So the peak of these things are representing beta. And the goal of inference, right, is to figure out what the pattern of those betas should be to minimize discrepancy between the predictable response and what we saw in the data. All right. And so these all have their associated HRS, which are those dashed lines there, and then we just need to convolve that signal. Now in a finite impulse example in discrete time, you can just sum these up. Really simple, you just add them up through there, and this will just give you this basic HRF, okay? And so that's really all there is to it. So you're describing now, the last part is just regression. So you have your mean prediction, which is this x times beta piece, and you have your neural data, so you'll filter that through there. The last part is just specify how much you missed. You need some probabilistic function, you know, if you're trying to estimate the parameters. So that's just specified by sigma. So there's a parameter for that that tells you, you know, how bad you're doing, basically, how far off are you. And so that's all there is to it. All right, so now we can move on to the decision component. So how do we make a prediction about which of these two stimuli this subject will say has the highest contrast? And so we're interested in both choice and RT. So a simple way of doing this, right, you might just say, oh, the neural activity, we just correlate it with RT, whereas, you know, you could also correlate it with choice. But the idea is that the joint distribution of choice and RT are very important here. And, you know, at least in the field of mathematical psychology, you know, the joint distribution of choice and RT has really helped to kind of tease apart model differences and give us more evidence for some architectures versus others. So both of these are important. There's a very long history about that, why that's the case. So here's the diffusion model. So we have, basically, you're representing the two choices as boundaries. So there's choice 1 on the top, choice 2 on the bottom, and time is just on the x-axis. Now, whenever you present these two, the second one comes on the screen, then they start accumulating information in this model. And so you're accumulating it, and these trajectories are supposed to represent that decision process unfolding with respect to time. And if it's a really easy difference, like a really easy trial, a very large difference between the two contrasts, you would expect them to make a decision right away, right? And so the model captures that by a mean trajectory parameter. And so all the parameters are listed here, and I have like a little description next to them. So trial-wise drift rates, drift rate is that rate of evidence accumulation I mentioned. So easier trials, it should be faster trajectory. In other words, it should be pointing like right toward one of the boundaries. The threshold parameter, the distance from 0, that's just called alpha. So it's the degree to which you need alpha information in order to make a response. And so that distance is alpha. Response bias is omega. So basically, you could start, for some reason, a sequential dependency or something like that. You could start closer to one of the bounds versus the other, right, than what like an unbiased observer would. And if that happens, you know, you can estimate that parameter, omega. And then lastly, we have a non-decision time. So this isn't a complete model, right? It's an abstraction, just meant to kind of describe how the two variables are related to one another. And so we have to build in other things. So we don't have things like visual encoding or the time, like after they've decided to make their response, they have to go press a button. So that time, those pieces are not captured in the model, right? So it's just a piece that is assumed to vary from trial to trial. All right, so that's the full model specification. And as I mentioned, there are these tutorials. So like this is the tutorial for getting the R-Wiener module into your system. You can just follow that. And there are also people that have done this within STAN, and that's well documented now as well. Okay, so for simplicity, did you want to take a picture of that? All right, so for simplicity, we're just going to focus on two types of linking functions just to be concrete here. So there's the neural response can be directed, or it can be this covariance structure that I mentioned. So just to summarize, you know, the neural model response, like what's going to basically predict that HRF is the difference between these. So the difference between these two beta estimates on every trial is the critical variable that's going to signal a change in the drift rate parameters, or the rate of evidence accumulation. And that's denoted by C. All right, so in the directed linking function, really simple. You just take the difference between them and you just say that's C. So you have C, and that will is input in here to the diffusion model process. These other parameters you can just assume they're fixed across trials, or you can play around with that. If you want to estimate those trial by trial, it turns out you can do that. So that's really handy. And you're explaining the joint distribution of choice, denoted C, and response time, RT, on every trial. Okay. So this little syntax that will define what that means in the model. And then lastly, the other variable that we obviously really care about is the neural response. We want to explain that time series as well. And we have ways of doing that I already told you about. Now for the covariance linking function, it's got the exact same kind of syntax here, but the way that we get a C estimate is we take this specification, these two parameters, and we're just feeding them through a multivariate normal. So here we only have one neural parameter of interest and one cognitive model parameter of interest, just to keep things simple. So this is just a simple bivariate normal. So that means we have phi, which is a mean for all of the drift rates and all of the bold responses across time. And then you have sigma, which tells us about the covariance and the variance of each of those parameters. So it describes the variance of the neural data, or the neural model parameters, the variance of the single trial drift estimates, and the covariance among those two parameters. All right, so here's how we model the bold response. And remember, I told you this was nested within the likelihood. So if you open up the JAG syntax, you get something that looks like this. So this big blob of code just does this convolution process. And so we have this where it's just a finite impulse response, but again, you can do better if you try this and don't like it. And then there's the HRF that we just programmed directly into JAGs. The prior, if you're working in a Bayesian framework, you need to specify prior distributions. This is subjective, so I'm not going to give you advice. It's the point is you have your priors and I have my priors, and some people don't like that, but I happen to. So you can specify these priors however you like. If you have a lot of information, you're like an expert DDM modeler, you can change this and make it much more precise. Here we chose to just be kind of agnostic, just to kind of protect us from any guilt of the model not fitting or something like that. Okay, so modeling choice response time, you've got your time. Remember, that's a vector, which is a little weird, but once you have that negative coding thing that I mentioned, you can just describe those t's as a function of this d Wiener function, which is the one that I told you they programmed up in C. So JAGS is somehow accessing that, and it's like an internal module now. All right, so that will give you the description of behavior, and the neural response is just there. You just subtract the two and create a new node in the graph. All right, the last part is the linking function. So here we have our likelihood function, we have our differences, and we need to define what the linking function should be. So here we're describing it with a multivariate normal. So it's a little weird, maybe this is not the best choice, but both the difference in the two neural parameters and the behavioral parameter are contained in the matrix called drift. That's maybe a little misleading because it's not really just the drift, but whatever. So we have drift I, and that's going from 1 to 2. So you're specifying a vector, it's two-dimensional, and it's going through this multivariate normal. Okay, and that's really all there is to it. Then you need your R handler code. So if you're trying to implement this, you're trying to run from one R script, and you don't really even need to look at the JAG syntax, but it's there if you want to check it out. And it's just a couple of things here. I'm not going to be able to tell you all about JAGs in this talk, but the basic idea is you need to adapt the JAGs model. There are ways of kind of getting your chains. Okay, so Bayesian sampling, you have to use chains in order to sample from a target distribution to get a posterior estimate. So the posterior estimate is akin to just a single point estimate, but the posterior estimate is expressing uncertainty. Okay, so it has both the mean, but also just the probability that any given parameter is producing the data that you observed. So now that you have that out of the way, we have these algorithms, we have to specify what those algorithms are. And JAGS has a lot of stuff that's internal that will just populate for you, so you don't really have to think hard about this. It's fairly robust, but it does kind of change how much time the computation will take. So if you're kind of on a budget in terms of like a deadline or something, you can specify how this works, a number of adaptations and chains and so on. And if you really are like, it's desperate to get the right answer, then you might want to beef this up. And so have more samples that you draw from the posterior. So the more samples, the better. And that's just a general case. Okay, so there are a couple of pieces. You have to initialize. You then do this thing called burn-in. So you need to draw samples that you don't necessarily think are the right samples. They're just algorithmic in nature. They're just kind of getting you in the right area. So this dimensional space is vast. It's very large, a sea of uncertainty, and you're kind of traversing this with the algorithm, but you have to first let the algorithm do its work before you can start actually reporting the results. So you just run this burn-in period, and that will get you to the right neighborhood, and then you can draw directly from the posterior. Okay, so just be careful with that. There are diagnostic tools for this called trace plots, autocorrelation functions, and things like that that you can do to protect yourself. All right, so the last part, the posterior sampling, and you can actually specify which parameters you care about. If you only care about drift rate, you can specify that and only draw samples from that. That's great for memory resources because sometimes these models get big and you don't want to be collecting all that. That creates a huge bottleneck and it takes a lot of time and is likely to cause JAGs to crash, in all honesty. So anyway, you can get all of these samples out, and there you go. There's your posterior. Now, in the code that I provided, we have forward-going solutions that will actually generate predictions for the bold response and allow you to compare, your predicted bold response to the data. It might look like something like this. I've got 2 different versions here, directed and covariance. In this small example, they're basically the same thing. There are some minor things that happen because the covariance one is protecting itself from variation in one dimension, whereas the directed is just susceptible to it. But basically, they're the same. And you can plot things like the drift rate estimate as a function of the RTs that were observed. And so like in a directed approach, you get this really nice prediction that, if the RTs are longer, you would expect the drift rates to be smaller, right? They're not processing, information as fast. So you get that estimate directly out. All right, the covariance thing, the only thing it really gives you over and above the directed approach, at least in this small example, is it gives you this pairwise correlation. So it tells you the degree to which V1 in this case is directly related to the drift rate. And so that shows up here, and this gives you a posterior. So this is unlike a credible set. You can just, we had an example earlier, I forgot whose slide it was, but basically you're looking at the posterior distribution relative to 0, right? A correlation of 0 means nope, nothing there. But since it's very highly concentrated in the upper end of that, it tells us that when V1 activity goes up, we should expect an increase in drift rate. All right, so all the code is there. If you go to the paper, it's much better than this talk. There are equations, there's code, there are really simple examples that are kind of, in my mind, they're kind of ridiculous, but at least they're like helpful in communicating this idea. And then there's some real data and the realistic application that I mentioned before. So I have, 14 and a half minutes. I was going to go through some of the code of the really simple example just to kind of show you what that looks like. I guess that laptop's back there, so I'll move back there. I'll try to speak loudly, but really this is just to kind of show you what the code looks like, and then this can be done. So if you have questions, feel free to ask and stop me if something doesn't make sense. All right, great. Yeah. Okay. So this is the directed approach. This is a simulation example. So it's very important that when you're trying to assess whether or not your code is working, that you do what's called the parameter recovery studies. The idea is you should establish that you know the true state of the world by picking some sets of parameters, and then you generate data, which is fake, obviously. Then you fit your model to that data, and you assess the posterior distribution relative to those true values. So here we go. If you scroll up here, this is not my computer, so I don't know if this is going to work. So here we go. So these things are specifying, first we have to load in some modules really quick. We have a couple of logit functions, which weirdly isn't internal to R unless someone knows that function. I've never run into it. The numbers model specifications, these are the number of trials we want to generate. The covariance for sigma 1, variance for sigma 1, variance for sigma 2, and the correlation between them. Once you have that, you can construct the sigma matrix, which is that, it's a big sigma, but it's lowercase here. Then you can specify the hypermean. Let's see. I don't have a keyboard, so that was a short sight. There should be a run. Yes. All right. So that'll just load that in. Then you specify your hypermean vector, phi, and you'll draw random samples from a multivariate normal. So this MV norm. You think I would remember? Yes, there it is. Okay. All right. So there it is. And here's the time sequence we want, as well as the standard deviation of the bold responses, the number of ROIs, just kind of doing some house cleaning. And we'll go all the way down to here just so I don't have to embarrass myself anymore. Here we go. So now we have, just to show you what this code is doing, we have in, which is creating that, it's a random draw from some HRF, and so it's just kind of creating residual noise according to sigma. Then we have theta, which is just this product that I showed you before, x times beta, which is that. And then we, on each trial, are just sampling from a binomial distribution. So this isn't the diffusion model. This is just a simple recognition memory task. And we're just saying, did you remember it or not? And so this R binom is telling us yes or no. And it's always conditional on theta. And the idea, it's pedagogical, right? But it's just the idea is that increases in, let's say, hippocampal activity should signal higher likelihood that you'll remember something versus not. That's just the basic setup. All right, so then we create this data structure. We've got everything there. I don't think that's necessary to go into. So then we have our JAGS model here. These are similar lines as what we saw on the slides. So we have the JAGS model specified. I'm just picking 4 chains. Just 4 chains, number of adaptations. This is just kind of tuning, kind of getting the chains in the right area. This is not actually sampling. It's a different algorithm. Then you adapt, and then you update. This is drawing posterior samples. And, oh. I didn't even notice the errors until I looked down. Oh, I see. All right, so this kind of output will show up. So it'll be initializing. It's got this really fun animation, I guess. And so this will just draw all of your samples. And then down here, we're going to do some parameter recovery plots. So, this is a little more than necessary. I just really like nice graphs. So if you go into this, the part that matters here is just plotting posterior distribution for beta. And this EV equals something. This is just the vertical line for the true value that we actually use to generate data. And we can do that for both of those regions. All right, so as soon as that's done, this is the last step that will show up. Yeah, so this will create, this is not my computer, so I feel a little uncomfortable navigating through her stuff. She's kindly generated this for me. So anyway, this will create a PDF. If you run this, just kind of go into your working directory, just pull it up, and it will show you this posterior plot. It will be a posterior and It'll have a vertical line, and ideally it'll be centered somewhere in the vicinity. Now, there's nothing that guarantees the true value will be right in the center, so that often makes people really question this whole thing. But the Bayesian central limit theorem, it just guarantees that if you draw enough data points, that eventually the posterior will center on the right value and have low variance. But you're not guaranteed that with one simulation study. So you have to do this a couple of times to really assess whether or not it's going to work. Oh, I can do it. Great. So it looks like that. So here's the posterior, here's the other one. Now, we induced different correlations, right? So we induced one that was high, 0.5, and one that was low, like 0.3. And so we get something like that. All right. And I'm not really sure how much time I have left. 3 minutes? Oh, 5 minutes, great. All right, so this is the same basic code. Like, it's meant to be very similar so that you can just kind of pull them up and just kind of compare line by line. So the idea is it's all the same stuff. It's just now in the covariance, you're just inducing that correlation among ROIs potentially. So you can run all of this, and it actually is pretty fast. Yes, go ahead, please. Yeah, it certainly is a problem in the directed approach. It is not necessarily a problem in this hierarchical design. So the idea is you're sort of conditionally independent. So the hyperparameters are separated from the data. They're only influenced by the data through parameter nodes like delta. So even though delta increases in size, it's really just articulating, this covariance structure. Now, you could also make the argument that this is like a good thing. Like, you want as much complexity from the data as you can. So if you have a bunch of neural data, you just have to structure the model to be a little bit better at capturing all the time series, right? So what you're describing is you're increasing the complexity of this, and so that should affect the other. But if you just increase the complexity of your neural model, that will protect you from any unwanted changes in your behavioral model. But yes, it is definitely the case that, like, you have an objective function that you're multiplying together, and it will preferentially fit to whichever side is not described as well, right? So in this case, the complexity is on the neural side, and so it will try and compromise the fits to the behavior at the cost of the neural data. That's definitely the case.
Could you implement this also somehow in thinking functions, by weighting the different likelihood
I would really not recommend doing that, just because, you can't weight the likelihoods like that. It seems like a reasonable, like, intuitive thing to do, but if you start preferentially favoring one thing or the other, then you're not really doing a full likelihood, like a theoretically right thing to do, to kind of weight likelihoods like that. Yeah, so in that case, you might have to do something like hidden Markov models to kind of separate yourself from the complexity. And that hasn't been done in this framework, but some of the John Anderson's work does that. So yeah, that's a good question. Well, this isn't working, so. I'm not totally sure why. Oh, right. I think, yeah, so this happened in the review process. Somebody didn't like our definition that this was a hierarchical model, even though that's what we've been calling it, and we changed it to covariance. But the student that was helping out on this project didn't change that line, but changed the name of the file. So if you just type covariance where it says hierarchical, this will connect you to that file. I don't have a keyboard, I would just do it for you. I guess I could scroll around and copy and paste like individual letters, but I don't think anyone wants to see me do that. So Yeah, so just change that, and then this will produce, some nice samples. It'll show you these joint distributions of the recovered and estimated. And if you find a case where you don't recover something, I would love to know about it, because that's a problem that I need to solve. So, all right, so thank you for your attention, and email me if you have follow-up questions. I'm happy to provide the paper or more code or anything, other examples that we've actually used those diffusion models in a very big paper where we did all combinations of ROIs. So much more complex examples are out there, and I'm happy to help direct you to. So thanks.
I would like to invite all of the speakers to the panel discussion. So please come back. In the table or here? I think the table is the best option. But yeah, you can sit there. So first of all, thank you for coming. And please, if you have any question, feel free to ask. I will start explaining about the slides. That is one of the questions you have here. The slides, they will be available. They have uploaded this morning just before the course. So I don't know when they will be ready, but they are there. So any questions? Or we'll start with the questions here. You first?
Yeah, so it's a little hard to get into details about your experiment just in this venue, but you definitely want to connect the neural data to the stimulus design, just because there's a lot of raw power, because ultimately the brain is seeing this and the inputs are directly from the stimulus. And there are these clear, stimulus response things that happen where, saliency of the cue or the information, the stimulus does provide a really clear signal that you need to capture. Crosstalk differences, and that's a big problem, right? Because the brain's processing information differently, so you can't just be super agnostic about it. For that, you need to have a theory about what the differences are in the stimuli or the task demands that would cause such fluctuations in the neural response before you start connecting it up to a decision model. But this is part of why I thought the abstractions of cognitive models are so important, is they just do that, right? So like a drift rate doesn't mean anything, it's all contextual. So if it's a lexical decision task, perceptual decision like random dot motion, it will absorb the patterns that we care about in the behavioral response just by virtue of its abstraction. So but you know, I do think the goal of at least cognitive science is to kind of relate or better articulate what is the drift rate, where is its neural origins and things like that.
I don't have anything to add.
So first I would like to thank all the speakers for the excellent talk. Second, I just have a more slightly bit more technical question. So in the last, I think, two talks, we've seen like two powerful methods. One from directional base, where we have this beautiful machinery, you know, we can from ECM, it works very well, combination pattern. The other is from the hierarchical Bayesian or MCMC, which you know sometimes works. My experience is directional base is fairly fast, and some it cannot always give you results, especially when you have a practical data where you don't run truth. And on the other hand, when we run MCMC, sometimes we wait for a week or even several weeks because the parallelization doesn't help you. You have to run single chain, maybe I'm wrong with that. But I guess my question is there like suggestions that kind of gave on what occasions people should use what type of method, in particular, mainly directional base and Monte Carlo sum?
So maybe I'll get a practical answer to that. So I suggest, practically speaking, I would start with VB first, and then whatever model you're doing, I would do a full parameter recovery analysis. So I would check that when I simulate lots of parameters, the VB estimates of these parameters are accurate, right? So that there is no non-line identifiability, either created by the structure of the generative model or by the inference itself. And if that fails, I would go for Monte Carlo.
Yeah, I guess just to add on that, I would say that patience is a virtue and the truth is worth waiting for. This is often what I tell my students because, I mean, yeah, variational Bayes, I definitely agree with Jian's point. So it definitely gets you, you know, good estimates very quickly. It's really powerful machinery. But there are some issues with like covariance structure. So in these models where you have like a lot of parameters, it's sometimes difficult, especially in like the stuff I was doing. Like it's really important to me to articulate how a brain region is fluctuating with a cognitive model. And so that's all about the covariance. And when you're doing VB, it's really difficult to get the covariance without a really high dimensional Q function or kernel function. There is some recent work by Michael Jordan, though, for doing these kinds of corrections. So if you're interested in that, I can tell you more about that, but that's a bit technical. But MCMC always works, and yeah, you're right about the chains, but there are parallelization methods. So like we use these algorithms called differential evolution. So it's basically a particle swarm optimization algorithm that's running, but it's corrected for Bayesian sampling. So it allows all the particles to communicate with one another in a way that's extremely efficient.
Is that in JAX already?
No, this is homebrew. So, yeah, sorry.
Any more questions? Oh, yep.
Same
Yeah, so you can actually quantify the degree to which neural measures are informing the cognitive model parameters. So I get your point that, you know, I think it was related to what the other guy said earlier, where there's a dimensional mismatch, right? But you can actually quantify the degree to which those dimensions matter. So you can test to see, you know, which ROI is affecting the model parameter. And we have this other study where, you know, I want to connect behavior, EEG, and fMRI all together, and you can actually ask questions like, well, which is contributing more to the correlated pattern that I see? Like, how informative is EEG relative to fMRI? And so then you can make better decisions about which modality you want to invest in given the costs.
Okay. Yep.
Maybe a suggestion for discussion is I'm an fMRI physicist and I do behavioral studies as part of my fMRI studies, and I think that it's totally important to integrate the behavior information with the actual functional information. However, today I learned I know nothing. I was really educated in that sense that I don't have a chance for me to integrate that in my studies, because it seems to have an extreme amount of complexity. And even coming from fMRI here, words like VBA have another meaning for me. You know, even there, there is an initial problem in communicating. And I think that maybe there should be, or that I appreciate, or no, not appreciate. I mean, it's both communities coming together at that point. But I think that it might be more, it might have more impact on the community if we could find another level of communicating with another at that point. Right? I see in my group we have statisticians, or in my research cluster we have statisticians doing learning models, and apparently no one wants to cooperate because the level is of people communicate on different levels. Maybe that's, I don't know how you can work with that, whether you can try to standardize ways of integrating functional or integrating behavioral functional, or if you can build something like standardized interfaces. It seems to me that a lot of ideas go into optimizing your model for your certain approach, but maybe a more generalized level would make it easier for people from other areas to integrate, find some common ground, and start on further from there. I don't know what the panel thinks about that.
I think there are some, at least, developed more, which make it easier for interface. One of them is one Brandon mentioned before. Is HDDM is a package which enables you with relatively low knowledge of like the internal mechanics to implement some of the models, and to implement them some of the more sophisticated methods of like, well, that one is actually specifically to drift diffusion model, but do you know any other ones which are like have like easy interface for novice people to get into?
Yeah, I mean, so I think it's coming. So Russ Poldrack is working on this, the BIDS architecture, that's bit data standard. I don't know if you know this, but he's moving that infrastructure into computational modeling. So if there's like a set of core principal models that could be implemented within a containerized solution, then everything's preserved across users, and it has a bunch of nice replication kinds of features. But so it's coming. It's really hard. If only Google could also do Google Translate for scientists, this would be great.
As someone who's relatively novice in this, I agree that there's a very steep learning curve in this type of, and it's really difficult to get into because there's not many standardized methods. You need to like go through this all by yourself. Jean, do you have any comments on that issue?
I think you just highlighted. I agree with everything you said, but you highlighted a difficult issue. I think no one really knows how to solve that. And probably it's a deep issue because most of the time, I guess, the questions that require relating brain activity and behavior and stuff, they are subtle on the cognitive level, for example. So there is no overarching framework, essentially because people have very precise questions, and that may not relate to each other. So that's the way it is.
Yeah, in a sense, I agree with you. I myself also run into that kind of questions. For example, if I were trying to do something else, for example, if I want to learn VBA, I probably want to start from the scratch. Fortunately, there are toolboxes and packages available these days due to the effort of various scientists. But again, coming back to the bottom of the question, if you're a novice, if you have no experience at all in any of the related field that you want to do research, it's going to be a big headache. But I also want to emphasize different expertise is like it's like the recipes for different chef. You know, every chef has his own expertise. If you want to learn to cook a new cuisine, of course you can get the recipe from the other chef. But getting the recipe is not guaranteed to give you the right dish eventually. Even if there are like streamlined protocols to tell you what to do and what not to do, my own experience is that you will have to have some sort of expert guidelining you through all the process to make sure things are correct. Because sometimes even a real expert, maybe his expertise is not good enough to safeguard him in another field. So, yeah, I mean, I wish somebody had a solution for that.
I would like to stop there because we need to go through the questions that people posted online.
Oh, okay.
So, first question is not addressed to anyone. So, why is encoding treated as separate from evidence accumulation? Surely they are related.
Is what separate? Sorry.
Encoding is treated as separate from evidence accumulation. I think that is for you, actually.
Okay, as a different parameter. Well, it's basic assumption of like evidence accumulation networks that evidence accumulation is related to the decision process. However, encoding per se is not, meaning encoding does not actually influence our decision process. Of course, it can be faster or slower. However, it does not influence the decision process. Well, that's the kind of core assumption of the model. I know, Brandon, you want to add anything to that? That's fine. Yeah.
Okay. Next question: How you compare model fit? Which information criterion is appropriate?
There's a lot of them. Currently, the two, well, I can give a kind of concrete answer. There are currently two which are considered the best. However, don't ask me for exact mathematical proof of that. But currently, the two that are considered the best are the widely applicable information criteria, the WAIC, and the LOOIC, which is the leave one out information criterion. These two are considered the most sophisticated in terms of, and the most universal one. Maybe, generally, you would like to, I know, do you have like any intuitive answer why are the best?
Why they are the best?
Yeah.
Well, my understanding is that because they sort of take advantage of the posterior distributions of the parameter space that you're trying to integrate over. I don't do a lot of Bayesian, but my understanding is that a lot of model comparisons, I don't want to offend anybody, but but my own intuition is that for a lot of scientists, before they start experiment, before they collect data, they already have a sort of model in their mind that they preferred. There are a lot of maneuvering room in terms of getting you to the model that you want to pick. So I try to focus on the commonalities of the different models instead of saying, oh, you know, this one is in particular is better than all the others, especially when people are comparing like 20 or 30 models all together. But that's my take.
Yeah, I mean, I would tend to agree. I think there's a lot of consistency among the model metrics. I mean, I've been trying to run many of those together in every analysis, and the degree of overlap is ridiculous. So I think it's really just a soapbox that people get on about, like, this is the best metric right now, but they basically do the same things. So as long as you're checking all of them, I think that's probably the best approach.
I agree again with everything that was said before. I would just add that if you are Bayesian orthodox, then there's only one metric you can use, which is the model evidence. But probably it would give the same answer that most of the other ones. That's it. That's my 2 cents.
Okay, one question for you, Jian. Why don't you estimate the hierarchical model by plugging M into the equation for Y? It's for I don't know.
I'm not sure I understand the question actually. The reason I played around with this equation was to make it very simple and efficient to process fMRI data, so that we could effectively use the SPM or any equivalent software package to do a mediation analysis, without having to require anything else but the core routines of the software. So you can play around with this equation in many ways, and I'm sure you can get computationally inefficient ways of doing the same thing, but I don't see why one should try.
Okay, last one, and we can go. So, well, it's more like a comment. Model inference means inference between models. For example, model selection. No inference within model. For example, parameter testing. Any comment on to that?
I didn't really
I can repeat again.
Can you say it again? Sorry.
Model inference means inference between models. For example, model selection. No inference within models. For example, parameter testing.
Yeah, thank you for this comment.
Well, I think we are out of time anyway, so thank you for coming.
Great.
Like, big clap here.
I am. I'd be really keen on discussing things and stuff. So yeah, I'd be happy to hook up and then spend some time so that you can show me what you're doing and we can chat maybe.